These videos are offered on a pay-what-you-like basis. You can pay for the use of the videos at my website. There's a link to my website in the annotation. The address is www.freelance-teacher.com/videos.htm. Or you can just use the link in the annotation. Thank you. What does diploid mean?
Diploid is just two split cells. It's just it's like equal to 2n. It's in the process of like mitosis, the end of it.
Okay, so let's clarify that. So the key thing is that it means diploid describes the cell.
Okay.
So it means a diploid is a cell.
Okay.
With 2n. 2n what? 2n is a number. It's the number of what?
No, chromosomes?
That's right. So what would haploid mean?
That'd be 4 chromosomes?
That's right.
Or that's, yeah. Okay.
It's actually, one moment, 1 second.
All right.
So you were saying, what does haploid mean?
So haploid would be 4.
You said 4?
Yeah, or would that be, yeah.
4 what?
4 chromosomes, or 4 cells.
Okay, that's something that turns out not to be correct. So let's talk about that. Haploid here means half.
Okay, yeah.
So a haploid cell has n chromosomes.
Okay.
It might possibly be 4, but that would just be a coincidence. It doesn't have to be 4.
Yeah.
Okay, so a haploid would be a cell with n chromosomes. So what's the relationship between these? Well, a haploid cell has only one version of each type of chromosome. Remember, and a diploid cell has 2 versions of each type of chromosome. Haploid would have one version of each type of chromosome, and diploid would have 2 versions of each type of chromosome. Okay, good. So what does diploid mean?
Well, it has 2 versions of the chromosome.
2 versions of each chromosome. So what's the chromosome number?
2n.
2n. And what does haploid mean?
Haploid would mean 1 version of each chromosome.
That's right.
And that would be just n.
N, good. So let's say we take a parent cell and it goes through mitosis. Okay. How many daughter cells would there be?
2.
From one round of mitosis.
Would there 2 daughter cells?
That's right. Okay. Would they have the same chromosome number as the parent cell? So would this cell have the same chromosome number as the parent cell, or a different chromosome number?
It would.
Same or different?
Oh, same.
Okay, good. Would it be identical to the parent cell or different from the parent cell?
Wouldn't that depend if it was... Oh, wait. It would be... I think it's different.
So let's talk about that. It turns out that mitosis simply splits the parent cell into 2 identical copies.
Okay, that's...
2 copies identical to the parent cell.
Yeah.
So that would imply they're identical to each other as well.
Yeah. So mitosis is generally asexual.
That's right. Although it doesn't have to be a form of reproduction.
Yeah.
It could also just be growth. For example, when you grow, you have to create new cells. And what's the name of the process that those new cells are created by? Mitosis. Okay. So if it's used for reproduction, it would be asexual reproduction. Okay. And it would then make 2 offspring that are identical to the parent, assuming there's no mutations. But if you had, if it was just growth, it would be making 2 daughter cells identical to the earlier cell to help the organism grow. So we have 2 copies identical to the parent cell. So what did we say? Does this have the same or a different chromosome number as the parent?
The same.
Same chromosome number as as the parent. So, if the parent is 2n, what would be the chromosome number of this daughter?
2n.
And this daughter?
2n.
But it's also possible to have mitosis when the parent cell starts with n.
Yeah.
In that case, what would be the chromosome number of this daughter cell?
N.
And this daughter cell?
N.
Okay, good. Okay, so if you do mitosis, how many daughter cells does that give you?
2.
2. And are they the same or different as the parent?
They're the same.
Okay, so now more complicated, let's talk about what happens in meiosis. So here's a parent cell. Now, in meiosis, the parent cell is always 2n.
Yes.
At least in the standard examples that you're working on right now. So the parent cell here now would be 2n. And so then how many daughter cells? So if we do a complete meiosis, if we complete the process of meiosis, after completing the complete meiosis process, how many daughter cells are there?
1.
This turns out to be 4.
Oh, okay. Okay, yes.
I think you probably studied this in the first semester. Do you remember there's actually 2 rounds, meiosis 1 and meiosis 2? So we don't need to go into those details now for these chapters, but we should know that since there's 2 rounds, there will be 4 daughter cells.
Yes, correct.
Although not always, some of them don't always survive, but we still need to know we are originally producing 4 daughter cells. Okay, good. And what will be the chromosome number of this daughter cell?
I think N.
And how about this one?
N also?
They'll all be N.
Yeah.
So the key point is, what does meiosis do to the chromosome number?
It cuts them in half.
That's right. What did mitosis do to the chromosome number?
It kept them the same.
So that's an important distinction. Good.
Yes.
How many daughter cells does meiosis have?
4.
And how many does mitosis have?
2.
All right, so that's also an important distinction.
Yes.
Okay, so now will these daughter cells be identical to the parent?
No.
They can't because they have a different chromosome.
Yeah.
And will they be identical to each other?
No.
No, because remember, each of them is getting, so there's going to be a different splitting up of these 2n chromosomes. Remember, this has 2 versions of each chromosome, but those are not going to be split up identically and identical patterns between these daughter cells. Okay, so the daughter cells are 4 non-identical cells. Okay, so when you're doing meiosis, how many daughter cells are there?
2. Wait, no, sorry.
For meiosis, how many daughter cells are there? 4. Good. And what does meiosis do to the chromosome number?
Cuts them in half.
Good. So what chromosome number would you start with for meiosis?
2n.
And you would end up with?
4n. 4n was like 4 singular.
Yeah, so the new chromosome number would be n, but there would be 4 of them. Okay, yeah. Maybe that's where you were getting, possibly you might have been thinking of that number 4 earlier. Okay, on the other hand, notice mitosis could start with 2n or with n. You could start with either. Meiosis always starts with 2n, but mitosis could start with either 2n or n and keep the same chromosome number. Okay, so now let's see how this applies to plants. Remember, in plants, there's what's called an alternation of generations. There's 2 different generations in plants. Do you know what the names of the 2 generations are in plants?
Would that be, I think it's like P1 and P2?
P1, I think that's a different context. So what I was going for here is there's the Sporophyte generation.
Oh, okay. Or was it, oh no, it was different. It was generation 1, generation 2. That was
you
just
might be thinking
Mendel.
Mendelian genetics.
Yeah.
That's not really directly related to this concept. Okay, so there's the Sporophyte generation. Do you remember what the name of the other generation is?
Um, s- no.
So those are two terms that could definitely come up on the exam. So this would be the
Yeah.
gametophyte generation.
Okay.
So what happens is a sporophyte organism gives birth to, so to speak, or creates the gametophyte generation. And then the gametophyte generation gives birth to, so to speak, or generates the sporophyte generation. So
So
there's
it's
really two different types of organisms going back and forth. Sorry, you were going to say?
yeah. So it's basically like a cycle.
It's like a cycle. That's right.
Okay.
It's an alternation or cycle of generations. So this is something that only occurs in plants. It doesn't occur in animals. A human being gives just birth to another human being of the same type. But a sporophyte plant creates a gametophyte plant, and then the gametophyte plant creates a sporophyte plant, and then the sporophyte plant creates a gametophyte. It actually alternates.
Yeah.
Okay, now how can we characterize these? What is the chromosome number for the sporophyte generation?
2N.
That's right. And what's the chromosome number for the gametophyte generation?
N.
Right. So these are both multicellular. This has a
Okay.
bunch of 2N cells, and this has a bunch of N cells. So you don't want to confuse a gametophyte with a gamete. A gamete is just a single cell, but this is not a gamete, it's a whole organism, a whole gametophyte organism, so it has a bunch of N cells. Okay, so that's the sporophyte generation and the gametophyte generation. Now, what you're focusing on in your exam is not all plants, you're focusing on angiosperms. Do you know what angiosperm means? What type of plant is that?
Is that a plant with both male and female reproductive systems?
Not necessarily. So
Okay.
angiosperm means flowering plant.
All right.
And that's the only type of plant we're focusing on in this chapter. So everything we talk about here will be about plants with flowers. So angiosperm is just a fancy name for a flowering plant. So how does the alternation of generations work for an angiosperm? Well, for an angiosperm, which is the major generation? When you see a flowering plant outside blooming, which generation is it in? Sporophyte or gametophyte?
S- Uh, could it be both?
If you're actually looking at a
Or
flowering plant, you're always looking at the sporophyte generation. So for flowering plants,
uh,
the sporophyte is the major generation. Which is bigger, the sporophyte or the gametophyte?
sporophyte?
Yeah. In fact, the gametophytes, remember I said the gametophytes are many cells, but actually they're only a few cells. So we're going to focus on this alternating generations for angiosperms. For angiosperms, the sporophyte is the major large generation.
Mhm.
The gametophyte is generally going to be relatively short-lived, relatively short. Maybe I shouldn't quite say that. It depends, actually. But this is going to be kind of the minor and very small generation. And we'll see more what that means as we go. So when you actually see a flowering plant outside, you're always going to be looking at the sporophyte. We'll see what we mean by that as we go. Okay, so to review, in the alternation of generations, what are the names of the generations?
Sporophyte.
Is one type, and what's the other generation?
Um, gametotype.
Good. Gametophyte.
Gametophite.
Sporophyte and gametophyte. Good. And what's the chromosome number for sporophyte?
2N.
And gametophyte.
N.
Good. And what's an angiosperm?
An angiosperm is oh, um, it's a flowering plant.
Okay, good. So for angiosperms, which generation is the major generation?
I'm sorry.
For angiosperms, which generation is the major generation?
Sporophyte.
Good. Which is the large one?
Oh wait, Um, the gametophyte.
Actually, that would be the sporophyte too. That's part of why it's
Okay.
considered the major generation.
Okay.
Okay, good. So those were these ideas that we talked about here. Sporophyte is the major large generation. Gametophyte is minor generation. There was an example testing those ideas on your previous exam, but that could come up again. Okay, so now we're going to go on and talk more. So again, what is a angiosperm?
The angiosperm is a flowering plant.
So we're going to be focusing now on flowers. What is a flower? Obviously, you know what flowers generally look like. You can see plants outside that have flowers, but what is the function of a flower in a plant? The flower is the plant's reproductive organs. The flower is the plant's reproductive organisms. So it's kind of analogous to the reproductive system of an animal. So the reproductive system for a plant is the flower. So we were talking about the flower. That's the reproductive organ for the plant, which is appropriate because we're working on the chapter on reproduction. So
Yes.
how many? So one thing that's definitely likely to be tested is the key parts of a flower. So what are the key? What are the key parts of a flower?
Like the major
Yeah.
primary ones. The roots, the stem, and the leaves.
Now, those would be key parts of the whole plant.
Okay.
Those would be the major parts of the whole plant, but the flower itself has 4 major parts. Can you think of some of the 4 major parts of a flower?
Like the petals.
That's one of them.
The stamen, the sepals, carpels.
Well, that's them. Good.
Yes.
So these are the 4 major parts of the flower: petals, stamen, sepals, and carpels. Okay, so we have to go through each of these and understand what they represent. So let's see. Okay, well, that's my attempt to draw some of the parts of a flower. So what would be the names of these elements, if this looks at all recognizable?
I'm not sure.
Okay. How about this?
Those would be the area of the carpel.
And this?
That would be the. Would that be the no? I know one. There's like 2 different sides. One's the.
So let's talk about this
carpel one.
together a bit. It might be a little bit difficult because my picture is not the best picture here. So generally, the standard flower has both male and female reproductive organs. So which of these is the male? Which of these is the male reproductive organs?
The steeple. Sepal.
Okay, actually that's the stamen.
Okay.
And the female reproductive organs are the carpels.
Okay.
That's definitely crucial for the next test. So here's the male and female reproductive organs. So obviously now we're seeing a big difference between the angiosperms and animals, because most animals are either male or female. Most animals don't have male and female reproductive organs in the same organism, but oftentimes flowers do. Okay. Now, so the petals and the sepals are kind of auxiliary elements. They're not the main reproductive elements. What are the petals? Well, you already know what flower petals are, right? They're just the main thing. When you think of a flower, you kind of think of the colorful, bright part of the flower, so you're usually thinking of the petals, right? So the petals are just the standard, colorful, bright part of a flower that people think of when they think that flowers are pretty. So that's what I was kind of, in a lame way, trying to draw here, the petals.
Yeah.
So this is oftentimes the colorful part of the flower. Why are petals colorful? What's the function of that color?
Well, to draw in light.
Okay, so it turns out that the function of the color is not to draw light, it's to attract animals.
Okay.
Especially like, say, insects, like bees or hummingbirds, or, well, I guess the color wouldn't help with a bat, but a bee, or it could help to attract a bee or some other insects or a hummingbird. These are often colorful to attract insects. And we'll talk later about why, or other animals. We'll talk later why they want to do that. Then the sepals, that's a part of the flower you oftentimes might not notice. That's these elements down here. Those are generally green, not colorful.
Okay. Yeah.
They look like basically little leaf-like things. One thing you're likely to be tested on is the functions of these elements. Do you remember the function of the sepal?
Isn't that what like before the flower blooms, it's what like covers the plant?
Excellent. Good. It protects the flower's bud. That's right.
Yeah.
Before the flower blossoms. Right. Or blooms. That's right. So this, the function of the sepal that your textbook covers is protects the floral bud. So before the flower had blossomed, all of this apparatus would be enclosed in a bud, which was covered by the two sepals. Okay. And then what's the function of the petals?
To attract insects or animals.
Yeah, that's right. And then the function of the stamen is male reproductive organ, and the carpel is the female reproductive organ. Okay, good. So what is the female reproductive organ?
Oh, wait. Yes, the carpel.
And the male?
The stamen?
Right, that's definitely crucial that we have to keep reviewing. Good. Okay, so furthermore, we're likely to be tested on the parts of each of those elements. So this element right here would be the stamen. The key part of the stamen is this part on the top. Do you know what the tip of the stamen is called? Or actually, maybe to back up for a second. So what is the stamen?
Is the male part of the
the male part. So what's its main function? What's the main function of the stamen in the reproductive process?
To release pollen.
Excellent. Very good. That's right. To release pollen. What's the main function of then the carpel?
Would that be the same thing?
So what would that be?
To release pollen.
Now, pollen is the male contribution, not the female contribution.
Okay.
Remember, is the carpel the male or the female?
Carpel is the female.
So that's going to be producing the egg.
Okay.
The pollen is going to be coming from the male, and the egg is going to be coming from the female.
All right.
Okay. So going back here, I don't remember. Did you know what the name of the tip of the stamen is?
No.
So this is where the pollen is released from. Do you remember what part of
Yeah.
the plant releases the pollen? That's called the anther. And that's another term that's very likely to come up on the exam. So let's make sure we label that. Anther. These names are kind of a pain to memorize because they don't, there's not any logic to it. It's just pure memorization. But it's very likely that lots of them are going to come up on the test. So this is important. So this is called the anther, this tip. The tip of the stamen is the anther. And then the lower part is called the filament.
Okay.
Okay, good. So what's the anther? Oops, excuse me.
Anther.
Yeah, well, go ahead. What's the anther?
Oh, anther is part of the stamen, and that releases the pollen.
Yeah, which part?
That
was maybe a weird question, but it's the tip.
Okay, all right.
What's a stamen?
The stamen is the male reproductive system
of the flower. Good.
Yes.
So here's what a stamen looks like. What do we call this part?
The anther.
Good. And what do we call this part?
The filament.
Good. What's the function of the anther?
To release pollen.
Okay, good. All right, so then continuing, this central structure here is going to be the carpel. I've only drawn one carpel, but some flowers have more than one carpel. But my picture is having enough trouble as it is, but many flowers have more than one carpel. So this structure here is the carpel. Okay, so there's three elements to the carpel. There's the tip, then there's this connection, and then there's the larger structure at the bottom, and we need to know all of those names. Do you know what this structure at the top is called?
Stigma.
Excellent, good. What's the key function of the stigma?
Oh, would that be like what filters the pollen that comes through the plant to for fertilization?
That's on the right track. That's on the right track. So let's clarify that. So the point is, remember, the anthers create pollen, but then the pollen is released from the anthers and moves from the anthers and moves over to where? It moves over to the stigma. Yes. We'll talk about how that works. So one way to remember this is stigma sounds like sticky, and the stigma usually is sticky so that the pollen will stick to it. Stigma sounds like sticky, and it is sticky because we want the pollen to stick to this. Remember, this is a sexual reproduction cycle, so we need, we need the product from the anther, from the male system, to make contact with the female system. Otherwise, we can't have the sexual reproduction. All right, and then this structure down here, any idea what this is at the base of the carpel?
Okay, that's where the fertilization happens. No, I do not know.
That's a key structure. This is the ovary.
Okay. All right, that makes sense. Because it's part of the female reproductive.
Right.
Okay.
So that would be a term that we use for the animal reproductive system as well. And then also we need to know the name of the tube that connects the stigma to the ovary. Any recollection of that?
It's not going to be the filament. It's not part of the...
So the filament is only for the anther. So this is what's called the style.
Okay.
Again, these names don't seem to make too much sense. They just have to be memorized. But they're definitely very important terms. All right, this is called the style. This is a tube that connects the stigma and the ovary. One thing that's confusing here then is there's three terms that start with ST. Three terms that start with ST. It would be easy to start confusing them with each other. So let's make sure we're not doing that. What's one term that starts with ST?
The stigma.
Stigma. That's right. What is the stigma part of?
The carpel?
It's part of the carpel, yes. And what's its function?
It's sticky and it allows pollen to stick to it.
Yeah, the pollen moves from the anther to the stigma. What's another term that starts with ST?
The style.
And what's it part of?
It is part of the carpel.
Good. And it just connects the stigma to the ovary. And what's the other term that starts with ST?
Stamen.
Stamen. Right. What's the stamen function?
The male reproductive for the flower.
Yeah. What are the parts of a stamen?
The anther and the filament.
So a stamen is kind of different because the stamen is one of the 4 key parts, where stigma and style are part of a carpel. All right, I don't know, you might remember this as stamen has the word men in it, and this is part of the male, man, so to speak, reproductive system. I don't know if that would help. And stigma is the sticky part, and I don't have any tricks for remembering the style, but anyway. Okay, good. So we have to remember not to confuse those 3 terms that start with ST. Okay, so these are the key parts, very likely to be tested on the exam. Very good. So what is the male reproductive organ?
Stamen.
Stamen, yeah. And what are its parts?
Anther and the filament.
Good. What does the anther do?
The anther is what releases the pollen into the air.
And what's the female reproductive organ?
Carpel.
And what are its parts?
Stigma, the ovary, and the style.
Good. And what's the function of the stigma?
The stigma is it's the sticky part of the carpel, and it's what allows the pollen to stick to it.
Okay, very good. Okay, so I think you're likely to see a question like this on the exam. So let's give this a shot. We need names for each of these elements. So what would the name of this be?
The anther.
Excellent. And then here.
That is the filament.
Excellent.
The sepal.
Good.
The stigma. Style. The ovary. That's basically the carpel.
Look more closely at what this arrow is pointing to.
Oh, the petal.
The petal. That's right. By the way, notice that the petals here are colored and the sepals are green. Okay, good. So what's the name of the structure that contains these 2 arrows?
Oh, the stamen?
That's right. And what's the name of the structure that contains these 3 arrows?
Carpel.
Okay, good. All right, so that would be a good thing to review for the exam. I think one of the objectives said there's a good chance that they were simply going to have you label from a diagram of an idealized flower, correctly label the following structures and describe their function. So it looks like there's a good chance you'd see that. One thing I should mention, what do we call the part of the stem that's attached to the flower? So this part of the stem that's attached to the flower, it's not really part of the flower, but there's a name that comes up in some context here. This is called the receptacle.
Okay.
So it's not really part of the flower, so I'll put like a barrier here, but this is the part of the stem that attaches to the flower. Part of the stem that attaches to the flower. Okay, very good.
Does it have any like actual functions, or it's just basically like a structure just to hold?
Well, it's a structure that's holding the flower. I don't see any particular reason why anything would be moving, too much would be moving from the flower into the receptacle. However, certainly it would make sense that there might have to be nutrients and water moving in this direction. So presumably I would expect most of the transport would move in this direction. The flower is going to need water and nutrients to support itself. So presumably its function is support and transport of nutrients into the flower. But those are things that actually are probably not going to be tested too much on the exam. We just need to know it's a structure that's connected, that connects the flower to the rest of the plant. Okay, good. So let's continue. Okay, so looking at your learning objectives, list 4 floral parts in order from outside to inside a flower. List 4 floral parts in order from outside to inside a flower.
Do they have to be like specific? Just anything.
Well, what are the 4 major parts?
The sepals, petals, stamen, carpel.
Good. I'm not quite sure what they mean by outside and inside. What would you say is the structure that's on the outside?
The sepal and the petal.
Yeah, I guess those are on the outside. I guess the sepal must be on the furthest outside, at least at the beginning, because remember, it encloses the floral bud,
True.
the floral bud. And then after it blooms, I suppose then the sepals and the petals are on the outside. And then I guess we would say that both the carpels and the stamen are on the inside.
Yeah. He said name 4 for inside and outside.
It says list 4 floral parts in order from outside to inside.
Okay.
4 floral parts in order from outside to inside. So I guess I would go sepal, then petals. And then who in my picture, who was in the center?
The ovary.
Yeah. Which structure is the ovary part of, though? We want to talk about the 4 major
carpel?
parts. Yeah. So I guess the carpel would be the 4th on the very inside.
Okay.
Right. So if we were going to go from outside to inside, what's on the far outside?
Sepal.
And then?
The petals.
And then?
No, not the reciprocal. The petals, the carpel, the stamen. Is that all?
So what was on the outside again?
The petals, the sepal, the carpel.
They said in order. So what? So first of all, what are the 4? What are the 4 major parts?
Okay, the sepals, petals, stamen, and carpels.
Sepal, petal.
Okay.
Stamen, carpel. If you're going to list them in order from outside to inside, which would go first on the far outside?
The sepal.
And then moving towards the inside, what comes next?
The petal.
I suppose so. Good. And then what?
Yeah. And I guess the stamen and then the carpel?
Yeah, after all, notice in my picture, I put the stamens on the periphery and the carpel in the middle. And I think that's generally how flowers are arranged. Usually the carpel is in the middle and the stamens are on the outside. So actually the order that we have them written here would be the best order, I think, from outside to inside. Mm-hmm. Okay. From a diagram of an idealized flower, correctly label the following structures and describe the functions of each structure. Well, we just did that exercise. Good. Okay, so now we have to continue talking about the actual reproductive cycle. So remember that we're talking here about an alternation of generations. Remember, what are the 2 types of generations for plants? They alternate between what 2 names of generations?
Oh, the sporophyte and the gametophyte?
Sporophyte and gametophyte. Which is the major generation that we see when we just look at a flowering plant outside?
Sporophyte.
So which generation is the flower part of? The sporophyte or the gametophyte?
The sporophyte.
Because that's the one that we see, obviously.
Yeah. So
the flower is part of the sporophyte generation. So what's the purpose of the flower? The purpose of the flower is to create the gametophyte generation. At least that's part of it. Now, but remember, there's going to be a male gametophyte and a female gametophyte. So first of all, we have to look at the creation of the male gametophyte. Okay. What is the male gametophyte? We briefly mentioned this before.
Stamen?
Now that's the structure that's going to create it. But what is the male gametophyte? The pollen.
Okay.
So what we're talking about here is basically creation of pollen.
Yeah, because the anther leaves. All right, got it.
We'll be more specific as we go. So where is the male gametophyte going to be created? Which part of the flower?
The ovary?
Now, we're talking about the male. Is that the ovary is part of the female system?
Yeah. Okay. So I was thinking that, like, the ovary is where fertilization happened.
Okay, good. That's a good point. But so the male gametophyte is going to come together with the female gametophyte for fertilization. But before we can get to that, we have to create the male gametophyte. So we've already discussed where is the pollen created.
The anther?
That's right. So this process is going to be occurring in the anther. So the cell that this starts with is called the microsporocyte. All right, this is simply one of the cells in the flower. So this is at the beginning here. So would you expect this to be N or 2N?
2N.
Right. Now remember, is the flower sporophyte or gametophyte?
Repeat that again?
Is the flower part of the sporophyte or the gametophyte generation?
Sporophyte.
Good. And what's the chromosome number for the sporophyte?
2N.
So, since we haven't made the gametophyte yet, we must still be 2N. Good.
Yeah.
Okay. Now, in order to create a gametophyte, we're going to have a meiosis step. So we're going to have this meiosis step. So how many daughter cells will there be from this step?
From meiosis, 2? No, 4. Sorry.
All right. Think about the review that we did for mitosis and meiosis earlier. So here we're going to get 4 daughters. Okay, good. And what is going to be each of their chromosome numbers?
N.
Right. So you learned in the first semester how meiosis goes through 2 cycles, meiosis 1 and meiosis 2. And at the end of that, it has created 4 daughter cells with half the chromosome number that we started with. Okay, good. We don't need to worry about all those details here. But anyway, here we're going to have those 4 daughter cells. These cells are called microspores.
Okay.
So notice it would be easy to confuse the microsporocyte with the microspores. So which is the parent? The microsporocyte or the microspores?
The microsporocyte?
Yeah, and which is the daughter?
The microspores.
Okay, that's right. I don't know, as a memory aid, notice this has the greater chromosome number and it also has more letters. This has a smaller chromosome number, so as the chromosome number decreases, the number of letters in the word is also decreasing. It's a little memory aid that we're going from the microsporocyte to the microspores. Ultimately, remember, these are going to be turning into the male gametophyte. And what is the male gametophyte?
The male gametophyte is just N?
That's the chromosome number, but what is it? It's going to be the pollen. So we're talking about how the pollen is going to be created. Okay, so we have these 4 microspores. So next, what's going to happen to a microspore? So we can take the microspore, and now there's going to be mitosis. 1 round of mitosis. What was the chromosome number for the microspore?
Just N.
Good. By the way, so notice that we are now beginning the gametophyte generation, right? Because what's the chromosome number for the gametophyte?
N.
So this is kind of the end of the sporophyte generation.
Yeah.
This is where the sporophyte generation ends. And this is the beginning of the gametophyte generation. It's a little tricky because these are not technically considered gametophytes yet. They're immature. But we're still at the kind of the beginning of what's going to create the gametophyte generation. Okay, so here we have the microspore, and we have to take these immature gametophytes and actually create mature gametophytes. So this is going to go through mitosis. So 1 round of mitosis would give us how many daughter cells?
2.
Okay, so that gives us 2 daughter cells. And what's going to be the chromosome number for this one?
N.
And this one?
N.
Because does mitosis change or maintain the chromosome number?
Maintains the chromosome number.
Yeah, so we're going to have the same chromosome number in both of these as we had before. In fact, so here we go. Now, we need to know the names of these 2 cells. So I don't know, do you have any recollection of what the names of these cells would be at this point?
Wait, no, those are microspores.
These are the 2 cells that are going to end up in the pollen grain. Do you know what the names of the 2 cells that end up in the pollen grain are?
Oh, no.
Okay, so let's go through that. One of these is called the tube cell.
Okay.
And the other is the generative cell. Okay. So there we go. And these would be surrounded these are going to be basically forming the pollen grain. So they'll simply be surrounded by the wall of the pollen grain.
Is there, or one of this, like, do they differentiate in size?
At this point, they would probably be approximately the same size.
Okay.
At this point, they might be about the same size. Actually, if I had to guess, I would actually think maybe the tube cell is probably bigger, actually.
Okay.
Tube cell is probably bigger, but that's actually not going to be too important for testing purposes. Okay. So now this is a little confusing. So these are also considered spores. Even though these are not the sporophyte anymore, the sporophyte generation is over, but the sporophyte generation has created these spores. You can kind of see that from the name, microspores. So actually this structure here is what we could call the spore wall. Which protects the spore. So here we have the spore wall, and basically this entire structure is the pollen grain. So at this point, the anther's job is kind of done. This is the process that takes place in the anther to create this. And so again, this whole thing, this pollen grain, this is the male gametophyte.
Okay.
Remember how we said that gametophytes are not just gametes. They're not a single cell. They're actually a multicellular organism. But on the other hand, remember, what did we say is the major generation, the large generation for angiosperms? Gametophyte or sporophyte?
Major was a sporophyte.
So which is the bigger one?
Sporophyte.
Yeah, and you can see that because here, here's the male gametophyte. It's the pollen grain. How many cells does the male gametophyte have?
2.
It's only 2 cells. Yeah.
Yeah.
So notice it's pretty small.
Yeah.
Okay. I think it'll get a little bit bigger as time goes on, but it's certainly very small. Certainly much smaller than the entire plant. Okay. Well, I think there's a good chance you might see a question that says, describe the process of the creation of pollen grains, or describe the process of the creation of the male gametophyte. You tend to have like short answer essay problems, right?
Mm-hmm.
So
yeah.
I think there's a good chance you're going to have to reproduce this process. So basically, we just need to stare at this for a while and try to memorize this. So what's another name for the male gametophyte?
Microsporocyte?
Actually, no. That creates the male gametophyte. What I was going for is the male gametophyte is the pollen.
Okay.
Or the pollen grain.
And that creates the microsporocyte?
Well, let's go through it again. So we have the creation of the male gametophyte. So what's another name for the male gametophyte?
Pollen grain.
Good. So what is the pollen grain? It's the male gametophyte. Where are the pollen grains created?
In the anther.
In the anther. Good. All right. So when we start this story, what's the name of the cell that starts the story?
The microsporocyte.
The microsporocyte. Good. We're starting the story with the microsporocyte. What's its chromosome number?
2N.
Good. Then what process does the microsporocyte go through to continue the story?
Meiosis.
Good. So how many daughter cells does that give us?
It produces 4 daughter cells with N chromosomes.
Excellent. Good. What are the names of the daughter cells of the microsporocyte?
Microspores.
Good. All right. And then what would, if we focused on one of the microspores, what would process would the microspore go through next?
The microspores go through mitosis.
That's right. So how many daughter cells would that produce?
2.
2, right? And their chromosome number would be?
N.
Good. What are the names of the 2 daughter cells?
I'm trying not to like look at the screen so I can
Good.
like pollen tubes.
You're on the right track.
Cell.
Tube cell. Good.
Okay, and the other one is generation cell. I had to look at the screen. I couldn't remember.
Okay, good. Something that so you mentioned what eventually is going to happen to the tube cell. What does it turn into? The tube cell, as you were kind of hinting, is going to turn into the pollen tube.
Okay.
We haven't gotten to that step yet, but maybe it's good to know already. The tube cell is going to become the pollen tube.
Mm-hmm.
And what is the generative cell going to become? Well, you should kind of be able to figure it out. It's going to become the sperm. Remember, since this is the male system, eventually we have to get the sperm.
Yeah.
So the generative cell will eventually become sperm, and the tube cell is going to become the pollen tube. So notice, maybe that'll help you to remember the names. Why is the tube cell called a tube cell?
Because it's what, I'm not sure. Because it's...
What's it going to turn into?
Pollen tube?
Yeah, so it's called the tube cell, not because it looks like a tube right now necessarily, because it's going to create the tube.
Okay.
And then the generative cell is going to create, I don't know, well, we'll just memorize that's turning into the sperm. Okay.
Yeah.
Okay, so what's another name for the male gametophyte?
Pollen grain?
Good. What cell do we, so if we're going to, where does the gametophyte created, the male gametophyte?
Oh, in the anther?
In the anther, that's right. So if we're going to describe that process, what cell do we start the process with?
Oh, the microsporocyte?
Microsporocyte. Good. And its chromosome number is?
2N.
And then what process does it go through?
Meiosis.
And what does that create?
4 daughter cells with N chromosomes.
Excellent. And their names are?
Microspores.
Good. The microsporocyte creates the microspore. Good. And then if we focus on one microspore, what would happen to it? What would be the process it goes through?
It goes through 1 round of mitosis.
Mitosis to create?
The tube cell and the generation cell with N chromosomes.
Each. Good.
Yeah.
Tube cell and generative cell. Good. And what do we call that structure that has the tube cell and the generative cell?
The spore wall.
That's what encloses it. That's right. So what do we call the whole kit and caboodle? The spore wall that creates the
pollen grain?
That's the pollen grain. Yes.
Okay.
So let's take another look at our picture here. So the tube cell and the generative cell and the spore wall. So then what's going to happen now next in the process? Well, remember what's going to happen to the pollen grain? It's going to move over to where?
Tube cell?
Well, no, the tube is part of it. Now the pollen grain is going to fly off of the anther. So, for example, it might be blown off of the anther by wind. And then what part of the flower will it land on?
The stigma.
The sticky stigma, yes. So that'll be the next part of the story. Because remember that now that the male gametophyte is kind of complete or getting towards complete, it has to move towards the female part of the plant to continue the sexual reproduction. Okay, so the next step is that this is going to, the pollen grain is going to leave the anther. Okay, All right, so why don't you stare at what I have on the screen for a few more seconds? Maybe take 30 more seconds. Take 30 more seconds to stare at what I have on the screen and try to make sure you have this memorized. Maybe I'll get this memorized because your test is pretty soon, right? It's on Tuesday. So just stare at this for 30 more seconds and see if you, and try memorizing this, and then we'll move on. I'll actually give you 30 seconds. Go.
Okay.
Okay. Let's move to your screen. Let's say that you had a test question on the exam. Outline the process of the creation of the male gametophyte in angiosperms. So let's try to write down what we would do there. So we won't write down a full essay answer, but what I'd like you to do is try to, from memory, to write down what I have on my screen. So remember, I had kind of like an outline of the whole process. So try to outline pretty much identically what I had down, what I had written down, and you could use that to create an essay answer. So try working that out.
Okay. So,
and of course, try to do that without looking at your notes if you can.
Okay. All right. So the male gametophyte produces a. Well, it starts off with a micro. I can't really write.
Why don't we? Good.
Sporocyte.
Good. Go back and double click on the paintbrush on the icon. Oh no. Do you see where the ruler is at the bottom? I think you want to make the stroke down from 3 to 1 or 2 so that your pencil is not as thick. Good.
Okay. And that makes it, this microsporocyte is 2N.
Great. Maybe let's go to 2.
Huh?
Stroke 2.
Oh,
it's a little 2 thing.
Yeah. Okay. And that goes through the process of meiosis. And that produces, hold on. This is in the way. Okay. And that will produce 4 daughter cells, which are called microspores. With N chromosomes each. And then the microspores will become the this is the gametophyte, and they go through mitosis, and they produce 2 daughter cells with N chromosomes each, and they are tube cell and the generation cell. The tube cell is...
That's good. I don't know whether your TA would be picky enough to take off credit for this, but the technical name is generative cell.
Oh, okay. All right.
So let's write that correctly for your notes. Generative cell.
All right. And tube cell would become the pollen tube.
That's right. Let's not write that down yet, but that's a good thing to notice. It will become the pollen tube. But to keep things simpler,
okay.
So you're right. The tube cell does turn into the pollen tube. That's a good thing to know, but that comes later, so we won't write that down yet.
Okay. And then these 2 cells are enclosed into I'm trying remember the word. Was it pollen? Was it pollen wall? No.
So that would be a fair name. It's called technically the spore wall.
Spore wall. Okay.
Remember that this is created by the sporophyte, so it's the spore. Just like the microsporocyte creates the microspore, the sporophyte creates the spore. Okay, good.
Okay. So, okay. That is... All right, that
And
would
what
be,
do we call the whole structure? What do we call the spore wall, tube cell, generative cell? What do we call the whole thing?
The pollen grain?
Good. Now, let's get rid of that equal sign because that makes it seem like the spore wall is the pollen grain.
Okay.
So you want to put a bracket. The pollen grain is the whole thing, the whole collection. And you can kind of visualize, I don't know, maybe you've seen pollen. So it would be a tiny little grain that contains these elements. So the pollen grain is the entire thing.
Okay. And I guess now I would mention that the tube cell would become the pollen tube, and the generative cell will become the sperm.
That's right. If you want to, you can write that down below, or you already have that elsewhere in your notes. Let's not put it right there. So later, the tube cell will become the pollen tube, and later the generative cell will become the sperm cell. So will become later.
All right. Oh.
I don't want to have something in your notes that you might, when you go, I want these to be notes you can look at later. So we should say it will become that later. This is not, so if they actually, it doesn't happen while it's still on the anther.
Sorry, I gotta redo this. Okay.
Good.
Should I put this as like... Okay.
I think that's good. Okay.
Yeah.
What's another way to describe the pollen grain? What's another term that we could apply to that pollen grain?
The gametophyte?
Yeah, male or female.
Okay. So could I put an arrow and say male or female gametophyte?
Well, no, that was a question. Is it the male gametophyte or is it the female gametophyte?
The male gametophyte.
Right. So maybe to make sure this is clear, you can say the pollen grain is the male gametophyte. At this point, it's probably still considered somewhat immature, but the pollen grain is the male gametophyte. So we can just write down the pollen grain is the male gametophyte.
Okay.
I just want to have something written down so that if you come back to this a couple days later, you'll still know what it means. So we can just say the pollen grain is the male gametophyte. Okay, very good. So I think we did a good job on that. I think you remembered all the key terms. The one thing you had trouble remembering was the spore wall. That was a little less important, though. Everything else was good. Okay, so if there was a question that said, outline the process of the creation of the male gametophyte and angiosperms, these would be the elements that you would want to use in your explanation. Good. So I'm going to go back to my screen. Obviously, the hard part is still remembering that in a few days when the test comes around, but now you have something to work on to review. Okay, so I'm going to go back to my screen. When you're ready, take your time.
Oh, sorry. Okay, ready?
Good. One thing I just wanted to mention, I think you probably already figured out. So remember that the microsporocyte creates 4 microspores. So this process that I wrote down here for 1 microspore, actually all 4 of them would be going through it. So how many pollen grains do you get? How many pollen grains do you get from 1 microsporocyte?
8. Yeah, what'd you get? Yeah, 8.
Let's talk about that a little bit more. How many pollen grains do you get from 1 microspore? How many pollen grains do we get from 1 microspore?
Oh, 1.
1.
Sorry.
1 pollen grain from 1 microspore. How many microspores do you get from 1 microsporocyte? How
4.
many microspores do you get from 1 microsporocyte, you were saying?
4.
Excellent. So how many pollen grains do you get from 1 microsporocyte?
4.
Yes, 4, not 8.
Yeah.
Good.
I don't know why I said 8.
Okay, yeah. Okay, good. So that's a good thing to clarify. So we only showed 1 microspore turning into a pollen grain, but in reality, all 4 of these would turn into pollen grains. Okay, very good. Continuing, what would be now that we've discussed the complete creation of the male gametophyte, what would be the next logical thing to discuss?
The female gametophyte?
That was a weird question, but you figured out what I was going for there. Yes. So now we want to figure out, go over the process of creation of the female gametophyte. What's the name of the male gametophyte in angiosperms?
Microsporocyte?
Actually, that's a mistake. The microsporocyte was just the first cell that was creating the male gametophyte. Okay. What do you call the final product of that process? What do we call the male gametophyte?
Pollen grain?
Yeah, that's actually something to really highlight in your notes. I think that's especially likely to be important on the test. We don't want to... what's another name for the male gametophyte?
Pollen grain.
That's right. Just want to make sure we're clear. So what's the name for the female gametophyte? That's actually called the embryo sac.
Okay.
The female gametophyte is called the embryo sac. Okay, so let's proceed here. We haven't talked about this yet, but take a guess. Take a guess what structure... Remember we talked about all the various structures in a flower. In what structure in the flower do you think that the female gametophyte might be produced? Well, what structure in the flower... In what structure is the male gametophyte produced? In what structure in the flower is the male gametophyte produced?
The stamen.
Good. Which part of the stamen?
The anther.
Good. You know, that's something we should have put in our answer here. I think you might have forgotten to put that. If this was an essay question, you would definitely want to mention that this occurred in the anther.
Yeah.
The graders really like to know where things are happening. So that's something to make sure that you put in your notes. If they ask you to outline this process, you'd really want to emphasize that the male process occurs in the anther.
Yes.
Okay, so continuing. Now where might we take a guess, where might the female gametophyte be created?
In the carpel.
Excellent. Which part of the carpel? Take a guess.
The ovary?
That's right.
Okay.
Now it becomes more complicated, though, because there are subcompartments inside the ovary. We haven't talked about those yet. We haven't talked about those, but do you remember what the name of the subcompartments inside the ovary is?
I know... Well, I know one of them is basically the egg and the sperm.
Okay, so let's talk about that. Now, those would not exactly be the subcompartments. What I was going for is the ovule.
Okay.
And that is a very important term. We haven't talked about that term yet, but that's a very important term in this chapter. So, which is bigger, an ovary or an ovule?
The ovary?
Yeah. So is the ovary inside the ovule, or is the ovule inside the ovary?
The ovule is inside the ovary.
Okay, so this is to say, inside the ovary, inside the ovule is where this process is happening. Some ovaries have only one ovule, some ovaries have multiple ovules. But anyway, the process is going to take place in the ovule, inside the ovary. So what cell does this begin with? Now this cell, it turns out, begins with the... Remember, what was the first cell in the process for the male process? What was the name of the first cell in this process for the males?
The first cell created was the microsporocyte.
Right. I wouldn't say it was created, though. It was the beginning of the process. So
Okay.
anyway, the first cell in this process is called the megasporocyte.
Okay. That's okay. So that's the beginning of
It's
the
the
process for
cell
the
that
female.
begins the process of creation of the embryo sac.
Okay.
So we're not going to talk about how this is created. We're just going to start with the megasporocyte. We have to start somewhere. That shouldn't be too hard to remember because there was a similar cell for the male process, microsporocyte. Do you know what micro and mega mean?
In terms of biology,
Or just in terms of Latin or Greek? What does micro mean and what does mega mean?
micro means small, mega means larger.
Yeah, so that's the distinction here. This is presumably a larger cell than
Oh,
the
okay.
microsporocyte.
Okay.
Okay. Now, think about the analogy with the male process. What would be the chromosome number? So this is going to be similar to the male process. So what would be the chromosome number for the megasporocyte?
2N.
Because that was the chromosome number for the microsporocyte. Okay, good. And so continuing the analogy with the male process, what process would we now go through? What does the megasporocyte, what's the name of the process that the megasporocyte goes through?
Meiosis.
Meiosis. Excellent. So how many daughter cells does that produce?
4.
That's right. 4 daughter cells. What is each of their chromosome number?
N.
Good. And by analogy, what would be a good name for one of these cells if you think about the analogy with the male process?
So megaspores.
Excellent. Good. So one megasporocyte creates how many megaspores?
4.
That's right. Okay. So far this is very similar to the male process, right?
Mm-hmm.
But now we have a difference because usually 3 of the megaspores die
Okay.
and don't continue. It's different for all different types of angiosperms, but the ones that your chapter is focusing on, usually 3 now die. How many does that leave left?
1.
Yeah. So now we're going to continue with the 1 remaining megaspore. By the way, notice that we changed here from 2N to N. Remember our alternation of generations. What are the 2 names of the generations in the plant life cycle?
The sporophyte and the gametophyte.
Sporophyte and gametophyte, yes. What's the chromosome number for the sporophyte generation?
2N.
And gametophyte?
N.
So which generation is this?
That is the sporophyte.
In fact, you can see, though, this must be the end of the sporophyte generation.
Okay.
Maybe I shouldn't call it the end, because obviously the plant still exists. The plant still exists, it's still a sporophyte, but this particular cell, after it goes through meiosis, is not a sporophyte anymore. It's not part of the sporophyte. So what generation would the megaspore be in?
Gametophyte? No, yes, I would. Yes,
Yeah.
I would.
So this is the beginning of the gametophyte generation.
Yeah, I was just thinking that, like, the pollen grain was the gametophyte, but, like, it's the second process.
Well, the pollen grain is the male gametophyte.
Yeah.
Now we're looking at the beginning of the female gametophyte. Maybe I
Yeah.
should specify that. This is the beginning of the female gametophyte. Now, this is still very immature. It's not a mature female gametophyte, but it's the beginning of that generation. So the microspores were the beginning of the male gametophyte generation. Do you remember but they weren't the mature male gametophyte. What do we call the mature male gametophyte?
Um,
What's the name of the mature male gametophyte?
Microsporocyte?
I think you're getting confused. That was this part of the process.
Pollen grains.
The pollen grains. Yeah.
Okay.
So I should write down here, this is the mature.
Did you, I'm not sure if you said that, did you say if it was fully mature, the pollen grain?
I will say that pollen grain is the male gametophyte. It's going to mature even more than this picture.
Okay.
But it's we can call it the male gametophyte already. Right? The terminology is a little bit loose. Okay, so in any case, continuing, here we have the one remaining megaspore. What's its chromosome number?
N.
Okay. Do you remember what happened to the microspore? What process did the microspore go through?
Mitosis.
How many rounds?
One round.
Only one round.
Yeah.
But here, the megaspore goes through 3 rounds mitosis. This is pretty likely to be on the test.
Okay.
So this is going to go through 3 rounds of mitosis.
Okay.
So let's work that out. Let's see, what did I want? So basically, here, the megaspore has 1 nucleus. After 1 round of mitosis, how many nuclei would there be?
2.
Good. After another round of mitosis, how many nuclei would there be?
3. Oh wait, would there be another 2?
So I'll use a dot for a nucleus.
Okay.
So if you do mitosis, how many nuclei does that produce after
2.
1 round?
Well, okay.
1 round of mitosis would produce how many nucleuses?
2.
Good. And then let's say that both of these also go through mitosis.
Okay.
If they both go through mitosis, what's the total number of nuclei after that?
4.
Because this one creates 2, and this one creates 2.
Okay.
And then if they go through if they each go through a 3rd round of mitosis, what's the total number of nuclei after that 3rd round?
8.
Right.
Okay.
8 nuclei. Notice, every time you do mitosis, the number of nuclei doubles. Every time you do mitosis, the number of nuclei doubles. Does that make sense?
Mm-hmm.
So if you start with 1 nucleus and you do 1 round of mitosis, how many nuclei do you get?
2.
And if you do another round of mitosis, how many do you get?
4.
And if you do another 3rd round of mitosis, how many nuclei do you get?
8.
Okay. So here I actually drew a picture of all the nuclei splitting, but it shouldn't really be necessary to go through all those steps if we just remember that the number of nucleuses doubles from each round of mitosis.
Mm-hmm.
So the megaspore here has 1 nucleus. After 3 rounds of mitosis, how many nuclei are there?
8.
Yeah, 8 nuclei. And what is the chromosome number? What's the chromosome number of the megaspore nucleus?
Just N.
So what's going to be the chromosome number of each of the daughter nuclei?
N.
Good.
So, okay, the megaspore ends up going through the 3 rounds of mitosis, and then it produces just nuclei?
Well, here's where it's tricky. If you remember, what is usually involved in mitosis? Usually after mitosis, So mitosis means that you're splitting the nucleus, right?
Yeah.
Usually after mitosis, if you remember from the first semester, after mitosis, there's almost always cytokinesis. Remember that cytokinesis splits the cytoplasm so you get two new cells. But technically, those are two different processes. Just because you double the nuclei doesn't force you to split the cytoplasm. So what we have here originally is 3 mitosis with no cytokinesis.
Okay.
So would you agree then that even though we're getting 8 nuclei, at first we're still only going to have 1 cell? Does that make sense?
Yeah, that makes sense.
Yeah.
Just that one cell contains 8 nuclei.
That's right.
Okay.
We used to have 1 cell with 1 nucleus. We did 3 rounds of division, but we didn't split the cytoplasm up. We didn't split. So remember in cytokinesis, the plasma membrane pinches off into 2 daughter cells. That didn't happen here, at least not at first. Does that make any sense so far?
Yeah.
Okay. And what's the chromosome number of each nucleus?
N?
Yeah, they're each N. Okay, but now, soon, new membranes will form to split this up.
Okay.
Then membranes form. Like so. So each of these gets its own cell.
Okay, all right.
And each of these gets its own cell.
Okay.
Let's draw it like this. Okay. But notice these two in the center don't get their own cells. They're both part of the central cell.
Okay.
Okay, so now we have, I'm counting 7 cells.
No, wait.
With 8 nuclei.
Okay, all right. That makes sense.
6 of the cells have 1 nucleus each, and the large central cell has 2 nuclei.
All right.
Okay. So which of these are most important? Most important is this cell here. This is the egg cell.
Okay.
Obviously, the key to sexual reproduction is forming sperm and egg cell. So here's where the egg cell is going to come from. Now, that's definitely something you could be tested on, most likely. So I'll call this the egg cell. Then these 2 cells off to the side are the synergids.
Okay.
These 3 on top are the antipodal cells. Anti, because they're on the opposite side from the egg cell.
Okay, so I'm a little confused. I just wanted to clarify what is that's just one entire cell?
This is I would say this is 7 cells. 6 small cells and 1 big cell.
What is it contained in?
What is this cell contained in?
Yeah, what
it's
what
contained
is
in the ovule.
Okay, all right. All this is happening. Okay.
So this is all happening in the ovule.
Yeah.
So this is what the megaspore has turned into.
Oh, okay. All right.
So remember what happened is that the megaspore was like this. One cell membrane and one nucleus. Then at first, the nucleus split 3 times, 3 mitoses into 8 nuclei. But at first, it didn't split the cell membrane. So at first, the megaspore just turned into something that looks like this.
Yeah.
And then, I'm going to actually draw the process, then new membranes formed like this. So this will be like a little video. New membranes formed like so.
Oh, okay.
To create this picture.
Got it.
So that's actually a fairly accurate representation of what actually happens.
Okay. All right.
Okay. Okay, so that gives us then this picture here. So we now have 7 cells. Oh, and then there's no particular name for the big cell that's left. But the textbook sometimes calls this the central cell.
Okay.
It's a large central cell. So how many nuclei does the large central cell have?
2.
That's right. How many nuclei does the egg cell have?
1.
Right. So I'm trying to use a notation. I'm using dots for nuclei and the lines for the cell membranes.
Okay.
Actually, your textbook would draw the picture like this. Oh, so I should say these 2 in the center here, these are called the polar nuclei.
All right.
Now, your textbook actually draws the picture like this. They draw the 2 polar nuclei like this. But the problem with that is that then these look like new cells. It's hard for me to show that they're just nuclei. So I'm going to draw them as dots to show that they're not cells, they're nuclei. But otherwise, this is pretty much the same picture as your textbook uses. Okay. Now, this whole, this is kind of complicated. This whole structure, the whole kit and caboodle, everything together, this is what's called the embryo sac.
Okay.
Now, I'm not quite sure how much detail your test is going to go into on this, so let me highlight what's most important. The most important thing here are the polar nuclei and the egg cell. I don't know whether they'd actually test you on the names of the antipodal cells or the synergids or not, but these are the names that are most important. The polar nuclei, the egg cell, and embryo sac. Those are crucial. And it's crucial to know how many nuclei are polar nuclei.
Okay.
How many nuclei are polar nuclei?
2.
And how many nucleuses does the egg cell have?
1.
All right, so those are the parts that I'm sure are going to be tested. The other parts, not quite so sure.
Yeah.
Okay, so any case, that's the process. So let's see. All right, so that's a little easy to get. So notice this turns out to be maybe more complicated than the male process.
Yeah.
But I think we're kind of done now because this is the female gametophyte.
Okay.
So that's the end of this process. The embryo sac is the female gametophyte. We could call this the mature female gametophyte. Okay, so let's review the process. Ready to continue?
Yes.
So this is the creation of the female gametophyte, which is called the embryo sac. That starts in the ovary, in a part of the ovary called the ovule.
Okay.
We begin the story with the megasporocyte, which is 2N. It goes through 1 round of meiosis to make 4 daughter cells, which are called megaspores. So the megasporocyte creates the megaspores.
Mm-hmm.
So far, it's similar to the male process.
Yeah.
And each of these are N. Now we start to see the differences. So first of all, usually 3 of the megaspores die. And there's only going to be 1 remaining megaspore from this cycle.
Yeah.
Maybe I'll cross these out to show that they're dying. And it goes through 3 rounds of mitosis. Not just 1 round, which is what we had up here. And it's a little bit complicated. At first, all that does is the first round would change 1 nucleus into 2. Then the second round changes 2 nuclei into 4. Then the third round changes 4 nuclei into 8. At first, there's no cytokinesis, but once we have the 8 nucleuses, as I drew, a bunch of new membranes form so that we end up with 6 small cells and then 1 large central cell. How many nuclei does the large central cell have?
2.
Good. The other cell whose name you're likely to be tested on is the egg cell. How many nuclei does it have?
1.
1. And what do we call this whole structure?
The egg or the ovule?
This is the embryo sac.
Oh, okay.
It's inside the ovule. Okay. And I guess there is something else I should continue with here. This remember that. So the embryo sac is like the pollen grain.
Mm-hmm.
It plays the same function for the female that the pollen grain plays for the male. Okay.
Yeah.
Remember that the pollen grain was created by was surrounded by a spore wall. So this is going to be surrounded by what are called integuments.
Okay.
And they're just like basically the kind of protection or covering.
Wait, and that's okay. Integuments. All right.
They're kind of like the spore wall for the spore.
Yeah.
They're the kind of protection or covering of the embryo sac. Okay. So we're going over the creation of the female gametophyte. What is the female gametophyte called?
Embryo sac.
Good. So where does that process occur? The creation of the female gametophyte.
In the ovule, inside the ovary. Well, in the ovary, in the ovule.
That's right. What cell are we going to start the process with?
The megasporocyte.
Good. What is its chromosome number?
2N.
And then what process does it go through?
Meiosis.
Good. So what does that create?
4 daughter cells with N chromosome each, but 3 of the daughter cells will die, leaving 1.
Good. And what do we call that daughter cell?
A megaspore.
Excellent. Good. Then what?
The megaspore containing just N chromosomes go through 3 rounds of mitosis, making...
Now it gets complicated.
Yeah. It doesn't necessarily go through cytokinesis, but it produces 8 nuclei. And then through that, membranes will start forming around the nucleus, well, the nuclei.
Mm-hmm.
There are 2 large central cells that aren't like they're both together in the center.
Central nuclei. Good.
Yeah. And then you have the egg cell with 1 nuclei. And then that is all inside the integument, which protects the embryo sac.
Excellent. Good. Yeah. So what do you call the whole structure?
The embryo sac?
Excellent. That's right. So what's the female gametophyte? What's the name of the female gametophyte?
The embryo sac.
Good. What's the name of the male gametophyte?
Pollen grain.
Yeah, those are 2 things very likely to be tested.
Yeah.
Good. Okay, so that's the creation of those elements. Very good. What was the name again of the male gametophyte?
Pollen grain.
Remember that the pollen grain had 2 cells inside of it. What were the 2 cells inside the pollen grain?
Oh, the tube cell and the generative cell.
Good. And later, we haven't talked about this yet, but later, what does the tube cell turn into?
Pollen tube.
Good. And the generative cell?
Sperm cell.
So we need to do a similar process here.
Okay.
So, no, actually, maybe we'll come back to that later. Okay, so why don't you take 30 seconds to stare at this. This is kind of more complicated, but try to memorize here what we have on the screen. It's so complicated I can't fit it all on the screen at one time.
Yeah.
But I'm going to give you a full 30 seconds to kind of stare at this and try to memorize this so that you can write it out in an essay problem. Because again, you might see a problem like outline the process of the creation of the female gametophyte. So take your time and think about that.
All right. Could you scroll down a little bit? Thank you. Okay.
Okay, feel like you got it?
Yeah.
All right, it's kind of complicated, but let's move to your screen.
All right. Oh, wait, where is it?
Maximize your drawing window. Okay. All right, so let's say there was a problem on the test, the essay problem outlined the process of the creation of the female gametophyte. So we won't write out the whole thing in words like you would on the test, but basically try to write down exactly what I had on my screen. Try to write down the basic outline that I had on my screen of the process.
Okay. So oops. Female. It's the embryo sac. Okay. And then it's inside. Oh. No, thank you. No, thank you. Inside the embryo sac is inside the ovary. Inside the ovule. I don't know how to spell. Ovule.
That's correct. Good.
Okay, and then the cell that begins is the meiosis. Just 2N. Goes through meiosis. Okay. 3 will usually die.
Good. For your notes, on that meiosis line, why don't you make that into a rightward pointing arrow, just to make it a little clearer what's happening. Good.
Okay.
Because the megasporocyte is becoming the daughter cells. Good.
And these daughter cells. Spores. Okay, then the megaspore. which is just N, goes through mitosis. 3 rounds of mitosis. With no cytokinesis. And then that produces 8. Okay, with N chromosomes each. Okay. Okay, I have to scroll down just a little bit. Okay, the 8 nuclei start forming their own membranes. Embryo. And the 2 central nuclei. This is called the egg cell. This is all around inside the tegument. Okay. Those I don't necessarily remember these.
Okay, and I'm not sure whether you would be required to know those names or not, but those are called
Okay.
the antipodal cells.
Okay.
So if you're like doing an essay answer, if you don't know the name, you just leave it out. I kind of doubt that they would specifically ask you what the antipodal cell is, but it couldn't hurt to get credit to know those. And those are called the synergids.
Okay.
Okay, good. By the way, the central, what you have labeled as the central nuclei, they're actually called the polar nuclei.
Oh yeah.
That's a more useful name. You can change what you have off. Well, yeah, you can call it the polar nuclei. What we said is that the whole central thing is called the central cell. The central cell has the polar nuclei in it. So off to the left, you can erase central nuclei and replace that with central cell. So how many nuclei are there in the central cell?
2?
Yeah, the 2 polar nuclei. Good.
Oops.
Okay. Well, I think that's good progress on some pretty complicated material. I'm going to go back to my screen. Obviously, you need to keep practicing that and make sure you don't get it confused with the male process. It's easy to confuse those.
Mm-hmm.
Okay, so what is the name of the male gametophyte?
Pollen grain.
Good. And how many cells does it contain?
2?
Yeah, what are their names?
Tube cell and generative cell.
Good. What's the name of the female gametophyte?
The embryo sac
Excellent. Good. Okay, so those are some of the key ideas. Where is the pollen grain created?
in the stamen
Good. Which part of the stamen, though?
of the anther.
We really want to focus on that. Yeah, in the anther. And where is the embryo sac created?
In the ovary.
Good. We'll have to
In the,
go
in
one
the
step further too, in the ovule. So that would be good to have a two-part answer there. In the ovary, in the ovule. Okay, good. What happens to the pollen grain after it's created in the anther?
Wait, like after all the processes are
Yeah.
processes are
After it's
finished,
been created in the anther, what will it do next?
it will either be released by wind or insects, and it'll end up on the sticky stigma.
Very good. Okay, good. So what we just went over is this table.
Mm-hmm.
All right, so notice development of the male gametophyte. First, you have, notice it's happening in the anther region. The microsporocyte creates 4 microspores by meiosis. Each of the 4 microspores creates the pollen grain, the male gametophyte, which has a generative cell and a tube cell. So we went over that process. Then the female gametophyte is being created in the ovary. Which part of the ovary? The ovule. Which cell do we start with? The megasporocyte. It goes through meiosis to create 4 daughters, but only 1 survives. And then it goes through the 3 rounds of mitosis to create the... So here's the 3 antipodal cells at the top, the 2 polar nuclei in the middle, the egg at the bottom, the 2 synergids, and then outside is the ovule, which is creating it. And of course, the ovule is inside of the ovary. So now might be a good time to study this diagram. I think these are a useful diagram, but I think sometimes there's so much information in the diagrams that students can't get their bearings and see what's going on. So my hope is that now that we've talked through it, now hopefully this diagram will make sense. And you can go back and study it and meditate on it a little bit. So going back, what is the male gametophyte?
Pollen grain.
Good. Now the pollen grain, remember, is going to move from the anther to the stigma. What is the name of the process? What's the name of the process by which the pollen grain moves from anther to stigma? That's an important term.
Did we go over it?
No, we haven't talked about it yet. I'm sure you've heard it in class, though.
Oh, is that germination? No.
This is called pollination.
Okay.
That's likely to be tested. So, pollination is when the pollen grain moves from the anther to the stigma.
Yeah.
It's kind of a logical name. The stigma is getting pollinated. Good, but there's so many terms it's hard to keep them all straight. So this is pollination. Good. How does the pollen grain get from the anther to the stigma? Name 3 methods by which the pollen grain can move from the anther to the stigma.
Insects, wind. We said in class water. He said, yeah, that's possible.
Yeah. If the plant was in the water.
Yeah.
So if the flowering plant is in the water, then the pollen could simply float from one flower to another flower, from an anther to a stigma. Okay. By the way, remember that normally it's going to move from the anther of one flower to a stigma of a whole different flower, right? Because the whole point is to mix the genes up. So it would somewhat defeat the purpose if the pollen went from the anther just to the stigma that's in the very same flower. In many cases, it's going to move to a flower that's in a whole different plant. However, self-pollination is possible. Sometimes the pollen does just move from the anther to the stigma in the same flower or the same plant. But kind of the standard case we're going to focus on first is when it moves to a whole different flower. So it could be floated there by water. But more important, you mentioned insects, but it could be other animals as well. So the 3 types are animals, wind, and water. There's a good chance that could be tested. In fact, I think that was already tested on the previous test. So animals, wind, and water are the 3 methods. So how does it work with animals? So for example, an insect might move might brush against the pollen and the anther, and then when it flies to another flower, it'll transfer it there. What attracts the insects to the flower in the first place?
The color of the petals.
Yeah, that's one thing. They're attracted by the petals, or maybe the smell. That's why flowers smell good. What did the insect? What benefit does the insect get though from moving to the flower? Well, remember the flowers generally generate maybe nectar, that nourishment for the insects. Also, the insects and animals can just eat the pollen directly. The pollen is already highly nutritious. So the insects come in to eat the pollen or eat the nectar. Even though they're eating some of the pollen, they're not going to eat all of it. Some of the pollen just accidentally brushes up against maybe the legs of the insect or the wings of the insect. And then when the insect flies over to another flower, while it's collecting nectar or pollen from the other flower, it's just going to accidentally brush off some of the pollen onto the stigma of that flower. Okay. Mm-hmm. So that's that general process. So now what we have is so the stigma is part of what part of the flower?
The style.
So no, so the stigma is at the tip. And what is this whole thing called?
Oh. That entire area is the carpel?
Yes.
Okay.
What's this structure called?
That is the style.
Right. And what's this?
The ovary.
Good. And what's inside the ovary?
The ovule.
Good. And at this point, remember what's inside the ovule now?
The egg?
Well, the embryo sac.
Okay.
And the egg is inside of that. Okay, good. Now, I'm going to draw the stigma as bigger than it really is, just so I have space. So now in the stigma, we're going to, in fact, I'm going to draw it even bigger than that, just so I have plenty of space. Obviously, it would usually be smaller. All right, so on the stigma, we have the pollen grain now. And what's inside the pollen grain?
The pollen tube? No, the tube cell and then the generative cell.
Tube cell and generative cell. Okay, now that the pollen grain is attached to the stigma, do you remember what did we say the tube cell was going to become?
Pollen tube.
So now's the time. So now the tube cell turns into the pollen tube. I'm not sure whether it's more accurate to say it becomes the pollen tube or it produces the pollen tube. But anyway, the tube cell is going to produce the pollen tube. And I guess be part of the pollen tube. Where will the pollen tube go? So the pollen tube now is going to start inching down the style. The pollen tube now is going to start moving, growing its way down the style.
Is it inside the style or on the side?
It's inside.
Okay.
So
and then it connects to the embryo sac.
It keeps moving down until it finally gets to the embryo sac. That's right.
Okay.
Because remember, we want the male gametophyte to meet up with the female gametophyte. Right now they're separated, right? The male gametophyte is up here at the top, at the stigma, and where's the female gametophyte?
In the embryo?
In the embryo, it is the embryo sac, but where is the embryo sac in this
picture?
In the
ovary.
In the ovary and the ovule. So you can see there's a problem, they're separated from each other. So like any good romantic comedy, we need to get the female and the male gametophytes united together. So the male gametophyte, you can imagine romantic music playing as the pollen tube grows down through the style to connect the male, so that the male gametophyte can move down to the female gametophyte. Okay, So, so the tube cell is creating the pollen tube, and now as that is happening, the generative cell now goes through another round of mitosis.
Okay.
So let's make one of the pictures that we tend to make. We have a generative cell. It goes through one more round of mitosis. What was the chromosome number of the generative cell?
N.
That's good. Good. So how many daughter cells will it have?
4. I mean 2. I'm sorry.
Because it's
2.
Mitosis, not meiosis.
Yeah. Mm-hmm.
By the way, remember that there's only one round of meiosis. Everything else is mitosis. One round of meiosis in the male process, and then everything else is mitosis. And there's one round of meiosis at the beginning of the female process, and then everything else is mitosis. That just might help you as a memory aid.
Yeah.
We did meiosis at the beginning of the male process and the beginning of the female process. Everything else is mitosis. So we have 2 daughter cells. What are the names of these daughter cells? The names of these daughter cells are very important. Do you know what the generative cell produces? We briefly mentioned it before. So these would be the 2 sperm cells.
Okay. All right. I thought the generative cell itself. Oh, it becomes.
Well, it does. It splits and becomes 2 sperm cells. So the generative cell went through mitosis and created 2 sperm cells.
Mm-hmm.
What's the chromosome number of each sperm cell?
N.
Okay. So what's the chromosome number of this nucleus?
N.
Good. And what's the chromosome number of the nucleus of this cell?
N.
So I'm going to put dots to show the nuclei.
Okay.
So there's a nucleus in a cell. Good. Okay, very good. And where are those sperm cells going to go now?
In the egg?
Well, what I was going for is they're going to move into the pollen tube.
Yeah, they're going to travel down the pollen.
So once pollination occurs and the pollen grain reaches the stigma, the tube cell starts producing the pollen tube. The generative cell splits into 2 sperm cells, and then they start moving down the pollen tube.
Mm-hmm.
Okay. What is pollination?
Is when the anther releases the pollen and then it lands on the stigma?
Yeah, so that second part in particular. When the pollen reaches the stigma, that's pollination. Good. Mm-hmm. So then we have the 2 sperm cells. The 2 sperm cells would be moving down this pollen tube, moving down the pollen tube till they get to the ovary. And what's their destination in the ovary?
The embryo sac?
Yeah, they're going to be moving towards the ovule and the embryo sac. So eventually, the 2 sperm cells are going to reach. So I'll write that down. The sperm cells move down the pollen tube. Eventually reaching the embryo sac. Okay. Sperm cells are going to move down the pollen tube. Eventually, they're going to reach the embryo sac. Good. By the way, so the pollen tube is growing through where?
The stigma?
It grows away from the stigma.
Oh.
Through what part of the plant?
The style?
That's right. It grows away from the stigma, through the style, towards where?
Hello?
Hello? Can you hear me?
Hello?
Hello? So we're having, we
now.
keep getting, is there static again?
Yeah, it just keeps like, I can hear you now.
All right. Is it okay right now?
Yeah.
Okay. So I was saying that the pollen tube goes through the style towards the ovary, towards the embryo in the ovary. Okay. So what the sperm cells move down the pollen tube. They eventually reach the embryo sac. Okay. So this is this sexual reproduction. So what's going to happen when the sperm cells reach the embryo sac? So that's when, I guess that's when the sex happens. So that's when the sperm cells are going to be fertilizing the female elements in the process. So how many sperm cells are there?
That are produced 2 sperm cells.
That's right. That's why this process is called double fertilization. Fertilization is when a male gamete merges with a female gamete. But there's 2 sperm cells, so they're going to fertilize 2 different things.
Okay.
One sperm cell merges with the egg cell.
Wait, okay. Just one of the sperm cells?
Yes, one of the sperm cells will merge with the egg cell.
Okay.
What's the chromosome number for the sperm cell?
N.
And what's the chromosome number for the egg cell?
N.
Good. So after they merge, what will be their new chromosome number?
Oh. 2N?
Yes. So this is fertilization.
Okay.
This is the main most important step. This is the key step in the sexual reproduction. Do you know what do you call the egg cell after it's been fertilized by the sperm cell? So how many cells will that create? That'll create one daughter, right? Because these merge together.
Okay.
So at first there's one 2N cell. And what's the name of this? This is the zygote. That's an important term.
That's what it produces. All right.
So it produces one 2N cell, the zygote. So what's the chromosome number of the zygote?
2N.
Good. Remember, there's such a thing as plants have alternation of generations. Remember, what are the 2 generations for plants?
What are the 2 generations?
Yeah, what are the names of the 2 generations?
Sporophyte and gametophyte.
Excellent. Which of those is 2N and which is N?
The sporophyte is 2N and the gametophyte is N.
So notice now we just started a new sporophyte generation.
Yeah.
This is
a
new sporophyte generation.
Yeah.
So we've kind of started taking things full cycle. Remember, which is the major large generation in angiosperms, sporophyte or gametophyte?
Sporophyte.
Right. So if you see a flowering plant outside, are you looking at the sporophyte or the gametophyte generation?
Sporophyte generation?
Yeah, I'd emphasize that with you a couple times, but now you can see what I mean. Because notice the gametophyte generation, the male gametophyte is just the pollen grain, right? The male gametophyte is just the tiny pollen grain, and the female gametophyte you never see at all. It's just the embryo sac, which only has, which is just seven cells. So you can see that the gametophyte generations really are much smaller than the sporophyte.
And like you said before, yeah, like you said before, the gametophyte create the sporophyte, and I didn't get that in the beginning, but I understand that.
And then the sporophyte will create new gametophytes, and then the gametophytes will create new sporophytes. It's an alternation of generations. But the gametophyte generations are very small. The male gametophyte only has 2 cells in the pollen grain, and the female gametophyte only has 7 cells. It's still different from animals. Animals don't have any gametophyte or generations. They just have eggs and sperm. But anyway, hopefully now that's clear what that meant. So this is now we've ended that gametophyte generation, and now we have a new sporophyte generation. What is eventually going to happen to this zygote? Then the new zygote is going to turn into eventually the embryo.
Mm-hmm.
And then eventually that's going to turn into the new plant.
Okay.
All right, so we'll just finish this off here. So just like it's like in animal reproduction. In animal reproduction, when the sperm fertilizes the egg, I guess you haven't studied this yet in the class, but in animal reproduction, like in human reproduction, the sperm fertilizes the egg, that forms a zygote, which grows into an embryo, and that eventually grows into a new animal, like a new human being. So here, this kind of analogous, the zygote turns into the embryo, and that's going to that'll become a seedling and then a new plant. Okay? And of course, the plant would have what chromosome number?
2N.
Because that's the main sporophyte generation is 2N. So all of these here would be 2N. So this would be a very, very immature sporophyte. And as it grows, it turns into a mature sporophyte. Okay, but remember, how many sperm cells were there?
2.
So we've only accounted for 1 so far, right?
Mm-hmm.
So what happens to the other sperm cell? So we have to say the other sperm cell, it merges with the 2 polar nuclei.
Mm-hmm.
That's why this is double fertilization. So this is the other fertilization. This is also called fertilization. So together they're double fertilization. And that's happening in the central cell, right?
Yeah.
So now, the central cell is, well, what was the chromosome number of the sperm cell?
N.
And what's the chromosome number of one of the polar nuclei?
N.
And what's the chromosome number of the other polar nuclei?
N also?
So what is going to be the chromosome number of the new central cell after fertilization?
Wait. The mature plant?
Not in the mature plant. No, remember this is a totally different process. So we said that one sperm cell merges with an egg cell to form the zygote. And then I said eventually that's going to turn into a plant. But now we're still back at the beginning of the story. While one sperm cell is merging with the egg cell, the other sperm cell is going to merge with the 2 polar nuclei.
Okay, that'll be 3N?
3N. That's something we haven't talked about before. That's called triploid.
Triploid.
Triploid, yes, triploid. So that's kind of unusual. Most of the time, most cells are either diploid or haploid, but every once in a while, you see a triploid. So this new central cell is actually triploid. Okay. And you can see how that's happening here if we go back to our earlier picture. So here was the egg cell. Remember, the egg cell had 1 nucleus. So when the new nucleus comes in from the sperm cell, how many nuclei will there be in the new embryo? Oh, that didn't make sense. But the egg cell has N chromosomes. And then when N more chromosomes come from the sperm, how many chromosomes will the embryo have?
2.
2N.
Yeah.
But the central cell has 2 polar nuclei.
Yeah.
And then when a third nuclei comes in from the sperm and merges with these, how many chromosomes? So this has N chromosomes, and this nuclei has N chromosomes, and this nuclei has N chromosomes. So the new central cell after fertilization will have how many chromosomes?
3.
3N.
Okay.
That's actually pretty important. It's not going to have 3 chromosomes. It's going to have 3N. You have to see the difference between those. Okay. So that new central cell is going to be triploid. Now, what's the function of that? Remember, what is the zygote going to become eventually? After it finishes maturing, what does the zygote turn into?
The plant.
Yeah, eventually it turns into the plant. So what the heck is left for this central cell here? What is it going to do? Do you remember what its fate is? Do you remember what it becomes?
Does it become the
This is a very important term. This becomes the endosperm.
Okay.
This is a very key idea. So this becomes the endosperm. And what is the function of the endosperm? The endosperm stores nutrients for the growing embryo. So the central cell is going to store nutrients. The central cell, which is 3N, becomes the endosperm, and that's going to store nutrients for the growing embryo. Okay, good. So what's the chromosome number of the endosperm?
2N.
Well, remember, where did the endosperm come from? It came from this central cell, which is 3N.
Okay.
So it'll still be 3N.
It'll okay.
Yeah. The endosperm will now, presumably, now the central cell will maybe continue dividing by mitosis. I'm not sure whether it does, but anyway, the endosperm, I don't know if it splits up into more cells or not, but anyway, it's going to maintain 3N from the central cell. Okay, so why is this called double fertilization?
Because both, like,
what are the two separate fertilization events? What happens in one fertilization event?
Once one sperm cell merges with the egg cell.
Good, to form.
And then that goes to fertilization to produce the zygote.
Good. Whose chromosome number is?
2N.
Good, and that eventually forms?
Into a mature plant.
Yeah, first an embryo, and then the new plant. And then what's the other fertilization event?
Is when the other, when the other sperm cell merges with the two nuclei cells, the pollen nuclei.
Right.
And then that creates a 3N diploid cell.
Triploid.
Oh, triploid. Sorry.
Good.
And then that goes through. Oh wait, that creates the endosperm.
Yeah, that is going to develop into the endosperm. Excellent. Very good. So what's the difference between pollination and fertilization?
Pollination is when the pollen is it's the process in which the pollen is attaching to the stigma.
Good.
And fertilization is when it's the process when after the pollen becomes the sperm, and then the sperm moves down the pollen tube to the embryo, embryo sac. That's when fertilization takes place.
And what happens in fertilization?
Double fertilization.
Yeah. What happens?
When one sperm cell merges with the egg cell
to form the zygote, and
yeah,
the other.
The other is when one sperm cell merges with the two pollen nuclei to create a triploid cell.
That becomes.
And then that forms into an endosperm.
Okay, good. So a common question is, what's the difference between fertilization and pollination? So it's good to see the difference. So when it's moving down the pollen tube, technically that's not fertilization, but when it reaches the embryo, so fertilization is when one sperm merges with the egg cell and the other sperm cell merges with the polar nuclei. Okay, and to finish off then, remember the embryo sac is inside of what?
The ovule.
Yes.
The ovule is inside the ovary.
Now, remember that the embryo, as the zygote matures, it turns into an embryo and then into the seedling. As that is happening, what does the ovule turn into?
Does the ovule turn into the seed?
Yes, very good. That's very important, often tested. As the embryo matures, the ovule becomes the seed. And what is the ovule inside?
I'm sorry?
What is the ovule inside?
What is... I'm sorry. The ovule is inside the ovary.
And as the ovule becomes a seed and the zygote becomes an embryo, what does the ovary become? We haven't talked about that, but do you remember that from class?
Does the ovary become... No. Does it become...
It becomes the fruit.
Oh, okay. It generates into the plant, okay.
Now, what's the purpose of the fruit? Well, remember that the plant wants to spread the seed out into the world so that the offspring can grow a distance from the plant. it doesn't want to just drop the seed necessarily right next to itself, because then it would have to compete with its own offspring. So remember, the seed contains the embryo that's going to turn into the new plant, but the plant doesn't just want to drop the seed right on the ground. It wants it to be carried away. Well, the fruit is tasty, right? So animals can come by and eat the fruit, and then since they've eaten the fruit, they've also eaten the seeds, and then they can fly away or move away. And later they excrete the seeds, and then the seeds have been moved to another location for the plant to grow up. Or sometimes the fruit is carried away by wind, depending on the type of fruit. So just like the plants need animals to move pollen from anthers to stigmas, they often also need to have animals eat the fruit to carry their seeds to another location as well. So the life cycle of the plants is very heavily intertwined with animals and insects in many cases. Okay, so to finish up and review here then, one of the sperm fertilizes what?
I'm sorry. Oh, one of the sperm fertilizes the egg cell.
That's right, and that creates...
That creates the 2N zygote.
Excellent, the 2N zygote, which
then
becomes
a mature plant.
Good. There's an intermediate step though. What do we call the zygote as it's maturing, as it's dividing?
I think I'm gonna remember. Hold on.
That's the embryo.
Oh,
that's an important term.
Okay.
So as the so while it's still in the seed, while it's still maturing, it's only an embryo. It's going to be a long time before it turns into the new plant. I just mentioned the new plant so you see what the ultimate destination is. But at first, the zygote is just an embryo. Okay. And then what does the other sperm cell fertilize?
It fertilizes, the sperm cell fertilizes the two polar nuclei.
Excellent. And that creates?
During fertilization, it creates a triploid cell, and then that goes to...
What does that become?
Goes the embryo. No, the endosperm.
Right. We didn't really say how it becomes the endosperm.
Yeah.
But it just becomes the endosperm. Good. And the triploid endosperm. What's the function of the endosperm?
Does it protect? No. Yeah.
Not protection, but storing nutrients.
Okay.
The endosperm is like the pantry. It's storing nutrients for the growing embryo.
Oh, okay.
Because it takes a lot of nutrition for this zygote to turn into a new plant, right? So one of the things that stores nutrients for that is the endosperm. All right. So in that growing embryo and endosperm, they're located inside what part of the plant?
Inside the ovary?
Yeah, inside which subcompartment?
The ovule.
Yeah, and what is the ovule going to turn into as the embryo is maturing?
The seed.
Yeah, and the ovule is inside the ovary, and what's the ovary going to turn into?
The fruit.
All right, so all that stuff we just covered is very important, very frequently tested. It's important to know the fates of all of those different elements. So the pollen grain lands on the stigma, the pollen tube growing down the style, and inside the pollen tube are the two sperm cells that we talked about. Remember, where did the two sperm cells come from?
The two sperm cells come from the male gametophyte, the pollen grain.
Yeah. What part of the
generative cell?
The generative cell. That's right. Remember, the generative cell did one more round of mitosis to make the two sperm cells.
Yes.
Where did the pollen tube come from?
The pollen tube came from the tube cell?
That's right. So remember that the pollen grain had a generative cell and a tube cell in them. After pollination, the tube cell turned into a pollen tube, and the generative cell turned into the two sperm cells. Now notice down here we have the ovary. Inside the ovary is the ovule, which contains the female gametophyte. You can see the integuments. You can see the two polar nuclei. You can see the egg cell here at the bottom. Okay, so then the two sperm nuclei move all the way down and around here. Here's the two sperm nuclei still moving through. And here they are. They're about to enter into the embryo sac.
Okay.
And then what do the two sperm nuclei do when they reach the embryo sac? Well, notice one of them moves into the egg cell to form the 2N zygote. And the other one moves up to join the two polar nuclei to form the endosperm nucleus, which is 3N. So that's the double fertilization.
Okay.
Do those pictures make sense?
Yeah.
Okay. So again, I just wanted to say, I know sometimes when people first see these pictures, it's hard to process the information. But now when you go back and study this, hopefully this should make very good sense. We basically talked about just about all the key elements of the growth of the pollen tube and double fertilization. So if you go back and look at this, this should make good sense to you now. Did you have a question?
Oh, no.
Okay. Well, looks like we're out of time. Okay. So there's a lot of really complicated information in this chapter. But we talked about some of the stuff there that was most important from some of the objectives there from that chapter. Okay, good. So that's our time for today. I sent you my tutoring policies, right?
Yeah.
Did you notice at the end there I mentioned most people don't actually benefit much from tutoring because they just forget what we went over in the session. So that's something you really have to worry about here. I think we made really good progress on this material today, but it would be really easy to forget it by the time the test comes around. After all, everything we talked about here is stuff you already heard from your class already, right? Everything we talked about today is stuff you've already heard from class or from your sections or whatever, but presumably it didn't stick those times. So it's really important to go back and really review everything that we just went through here. Notice what I kept doing was after we went through the mail process, I asked you to actually write down the mail process on paper, right?
Yeah.
That's the only way to tell if you have something memorized. So you should do that on your own. Go back and rewrite this male process on paper. And then you have to go back and rewrite the female process on paper. And I would have asked you then to write down the process for double fertilization on paper too. We just didn't have time. But you should definitely do that on your own. Most people, when they're reviewing, they just look at their notes. But that's useless. Just looking at your notes doesn't do any good. You have to take a blank piece of paper and try to write down all the main ideas that we just talked about on paper, and then check your notes and make sure you got it right. And you have to do that every day if possible between now and the test because it's just so easy to start forgetting this material. So that's kind of my homework for you. Write down the male process, the female process, and the double fertilization process every day until that's easy and those ideas are not getting confused with each other, because you can see otherwise that material doesn't stick. You'd already heard all this material from your class lectures, but I don't think a lot of this was stuff you hadn't remembered. So the only way to really the purpose of the tutoring is to give you the information in an organized fashion so that then you can practice it on your own. But unless you find the time to practice it on your own, you're not going to remember that material. Like I said in the tutoring policies, I always begin each session by quizzing the student on the previous material to make sure they haven't forgotten, because it's pointless to go on to new material if we've forgotten the old material. And I'm not going to, my policy is I won't move on to new material unless the student shows me they remember the old material. So if Monday comes around and you've forgotten a lot of these ideas, we're just going to review this stuff until we have it down again. There's no point going on to new material if the student has forgotten the old material. Now, obviously, it would be more in your interest if we don't have to spend too much time reviewing on Monday because there's so much other stuff to go over. It would be to your interest to review on your own so we don't have to review too much together. So what I'm saying is try to make sure you review on your own so we don't have to spend too much time reviewing together, so we can spend as much time as possible on Monday going on to new material. So like as we discussed last time, my policy for the tutoring sessions is that it's important to quiz the student at the beginning of each session on what we've covered, because it doesn't make sense to move on to the new material unless we know we have that old stuff down. So let's start by checking that. Last time we talked about the male gametophyte in plants. So can you tell me what is the angiosperm male gametophyte? What's the name of the angiosperm male gametophyte?
Sporophyte.
Take your time. That actually was incorrect. So, we want the angiosperm male gametophyte.
Pollen grain.
Okay, that's better. So try drawing the structure of a pollen grain.
Okay.
We just have to show the key elements.
Is that it?
That looks good. Good. For your notes, let's label that as the pollen grain, the male gametophyte. Okay, excellent. Last time we went over the whole process that creates the pollen grain, but to save time, we won't go over that today.
Okay.
Now, let's move on then to the female gametophyte. What is the name of the female angiosperm gametophyte?
Oh, the embryo sac?
That's good, yes. So let's draw a picture of the basic structure of the female gametophyte. So we were going to draw the basic structure of the embryo sac.
Yes. All right, it's the 8 nuclei. Just close.
Legal marijuana in Germany.
Oh.
Oh, man.
How's that?
Done?
Yes.
Okay. Okay, good. That's a good process. So let's just clarify a couple of things. Up at the top, you have those 3 cells at the top. That should be antipodal cells. Let's spell that correctly for your notes. So it's A-N-T-I-P, but erase what you have after the P so we can spell that correctly for your notes. So it's P-O-D.
N-T-L-P-O-D?
Yes, A-L. So that's correct in your notes. Good. Okay. And I think you have something pointing to maybe a central membrane.
Yeah.
Okay. However, it doesn't quite make sense to say that that whole space is a membrane. A membrane is a surface, not a space. So what we're trying to point to there is the central cell.
Yeah, correct. I'm sorry. Yeah.
Okay, very good. The 2 cells surrounding the egg cell are the synergids, but you don't necessarily have to write that down. I'm not sure if you're actually going to be tested on that. You've labeled the key parts, so that's good. Let's write down that the embryo sac is the female gametophyte for your notes. And, we might as well put that in. So let's label those two cells. I'm sorry, you can write the female gametophyte, and let's label the two cells as the synergids. This is an important structure, so we should know all the names. The two cells to the side of the egg cell are the synergids. SYNERGID. Okay, those are the synergid cells. Good. Okay, overall, that was good. This is a complicated structure, so that was a good process. What is the chromosome number of the egg cell? What's the chromosome number of the egg cell?
3N.
What was your answer? What's the chromosome number of the egg cell?
I said 3N.
Do you want to change your answer or stick with that?
Hold on. Oh, wait. Is it just N?
It's just N. That is something that is very likely to be tested. That would be a good thing. So last time we went over the whole process that creates the female gametophyte. I'm not going to go over that with you again. That would take too long, but you might want to go back and review that. That's very likely to be tested. So let's label next to the egg cell nuclei that is N. And if you go back and review the process, you'll see how it became N. What is the, point to one of the polar nuclei, please. Point to one of the polar nuclei. Good. What's the chromosome number of that polar nucleus?
N.
Good. And the other one is also N. In fact, all of those nuclei are N. All of those nuclei are N. The ones that matter the most are the egg cell and the polar nuclei, so you might put in N. You might as well put in N for all of those dots. That's actually one of the important aspects of this picture, is to keep track of the chromosome number. So that means, are they haploid or diploid or triploid?
Are they haploid?
N always means haploid.
Yeah.
N always means haploid. Okay, that's good. So there were a couple issues there. That would be a good thing to review before your exam. Let's move back up to the male gametophyte for a moment. That's good that you saw that had a tube cell and a generative cell. You can see it's tricky to scroll without zooming, but that's good. What is the chromosome number for the tube cell? What's the chromosome number for the tube cell?
Just N.
So let's write that in. And how about the generative cell?
N.
Yes, let's write that in. That's kind of the definition of gametophytes. Gametophytes are always N. So are they diploid, triploid, or haploid?
Haploid.
That's, again, part of the kind of the definition of the gametophyte generation. So we should add that because that's always important for exams on this topic. Okay, so...
should I write gametophyte is always N?
If you like, that's fine. That's kind of the generation of the gametophyte generation, and the sporophyte generation is always 2N. Okay, so last time we went over how these two male and female gametophytes were formed. That would be a good thing for you to review, but we won't spend time on that together right now. But what is pollination?
Pollination is the process where the pollen grains are sticking to the stigma.
Excellent. Where did they come from before they were on the stigma?
The anther.
Good. So when they've moved from the anther to the stigma, that's pollination. Excellent. All right, so what happens after the pollen grains stick to the stigma?
They move down the... forgot the name. Oh, the style. They move down the style into the embryo. I mean, the ovary. Good.
That's good. Those are important. That's very likely to be tested. Can you be a little bit more specific about the process by which they move down? Who do you mean by they?
Oh, the pollen grains. They flow down the style into the ovary, into the...
How so? Can you give a little bit more detail about that process? And what we want to do here is review some of the details we talked about last time.
Okay. How exactly? I'm not entirely sure. I'm not sure exactly how they travel down the style.
Okay. So let's review some of those key things because those are pretty likely to be on your exam. The key thing is, remember, what's the role of the tube cell? What does the tube cell become or create?
The tube cell becomes the tube... I'm forgetting the name. Pollen tube.
That's right. So that's one of those details that I was wanting us to mention. How is it possible? So once the once they are sticking to once the pollen grain is on the stigma, the tube cell creates a pollen tube that inches its way down the style. So that's one of those important elements of that. The tube cell is going to create a pollen tube that is going to inch down the style. Okay, so what's the role of the tube cell? It creates the pollen tube.
Mm-hmm.
What's the role of the generative cell? What does the generative cell do?
The generative cell becomes the sperm cell.
How many sperm cells?
2.
Yeah, what's the process by which the generative cell becomes 2 sperm cells? What's the name of that process?
Oh, the process, fertilization.
Actually, that wasn't what I was going for. Is it mitosis or meiosis?
Oh, mitosis.
That's right. So what's going to be the chromosome number of each sperm cell?
No. N.
Good. Because the generative cell is N, and as we talked about last time, mitosis always maintains the same chromosome number. Okay, so to review, what's going to happen to the generative cell?
The generative cell is going to become a sperm cell and go through mitosis. And it'll create 2 cells that have the chromosome N.
Right, so the generative cell goes through 1 round of mitosis. It splits into 2. And
Yes.
what are the names of the 2 daughter cells of the generative cell? Just to review what we were just talking about.
Yeah.
The generative cell goes through 1 round of mitosis. That means it splits into 2 new cells. And what do we call the 2 new cells that it forms? Those are the sperm cells.
Yeah.
Those are the sperm cells. So the generative cell goes through 1 round of mitosis to create 2 daughter cells. And what are the names of the 2 daughter cells of the generative cell?
I don't remember the 2 names.
Oh, I'm sorry, that's what I was just mentioning. Those are the sperm cells. Looks like we're getting a little confused.
Yeah.
But let's take our time on that idea. Maybe I'm going to move back to my screen.
Okay.
So the generative cell is going to go through a round of mitosis and form 2 daughter cells. What are the names of these daughter cells?
Sperm cells.
Right. 2 sperm cells. Good. What's the chromosome number of the generative cell?
N.
And of the sperm cell?
N.
Good. Okay. Now, after these 2 sperm cells have been created, what are they going to do?
One of them is going to go through fertilization.
Good. Now, let's try to trace that out a little bit more. The next thing they're going to do is they're going to move into the pollen tube.
Yes.
That was created by the tube cell. So remember that at first, these are both on the... At first, the 2 sperm cells are on the stigma at the top of the carpel. But the tube cell has created a pollen tube. The 2 sperm cells are going to move into the pollen tube and then move down the style. So... This is not to scale, but just to show us the basic parts. Okay, so the pollen grain is up here. The pollen tube is forming and moving down the style. And then the 2 sperm cells will also be moving down. The 2 sperm cells will move from the top down through the pollen tube towards the bottom. Okay, what do we call this organ at the bottom of the style?
The ovary.
Good. Okay, so to continue, what's going to happen when the sperm reaches the ovary? What happens next when the two sperm cells reach the ovary?
They move. They move to the ovule, into the embryo sac.
Excellent. That's right. What do they do once they've reached the embryo sac?
They go through the process. No, wait. They go through the process of meiosis, creating. It becomes a megaspore and it goes through the process of meiosis.
Okay. All right. Well, basically, what's happening here is that we're getting very confused. It looks like you're really confusing a lot of the different ideas from the previous session. The megaspore actually was involved in the creation of these gametophytes at the very beginning of the process. All right, so I'll help you a little with this. What actually happens when the two sperm cells reach the bottom is, so what is in the ovary? So they're going to go to the ovary. They're going to go into ovule. What is living there in the ovule? Who's already in the ovule when the sperm cell gets there?
Okay, the embryo sac.
The embryo sac is already down there. Remember my little cheesy romantic comedy analogy I gave last time? So the male gametophyte starts up here. The female gametophyte is down here. And then we can imagine the romantic comedy music playing as the male gametophyte sperm are moving their way down the pollen tube. Their goal here is to reach the female gametophyte for the sexual reproduction to occur. Okay, so the two sperm cells are moving down here. They're going to reach the embryo sac. So once they reach the embryo sac, one of the sperm cells is going to merge with the egg cell. That's what I was hoping we were going to mention a moment ago when I asked what was going to happen down there. One sperm cell is going to merge with one of the egg cells. The name of this process is not meiosis. The name of this process is fertilization.
Yeah. Okay. All right. Now I'm remembering.
The meiosis occurred much earlier in the process. Okay. So they're going to have fertilization. What's the chromosome number of this sperm cell?
3N.
So the question is, what is the chromosome number of the sperm cell?
Just N.
And the egg cell?
N.
Good. Now, do you remember, what do we call the product when the sperm cell merges with the egg cell? Once they've merged together, they form a new cell, and what's that new cell called?
The endosperm?
This is actually called the zygote. That's actually really important. This material we're going through right now will definitely be on the test. If we can learn this material here, that's points in the bank for us. All right, so this is the sperm cell, the egg cell. They merge to form the zygote. What's going to be the chromosome number for the zygote? This is something we can figure out from the equation.
2N.
Yeah, because you can see n plus n would be 2n. So this must be the beginning of the new sporophyte generation, as we discussed last time.
Okay.
Because remember, as we were talking about earlier today, the sporophyte generation is always 2n, and the gametophyte is always n. So since we've created a new 2n, this must be the beginning of the new sporophyte. Generation. Okay. So that's the zygote. What will the zygote eventually go on to form?
The embryo.
Excellent. Excellent. And what does the embryo eventually go on to form?
The plant.
Yeah, it's just the new plant. Okay, the new sporophyte version of the plant. Okay, good. What would be the embryo's chromosome number?
2N.
And the new plant's number?
2N.
Yeah, it's going to stay 2n for that portion of the cycle. Okay, like I said, this is points in the bank if we can learn this, because this is definitely stuff you're going to need to use in some form on the test. The sperm cell plus the egg cell goes through fertilization. This is the sexual reproduction right here, the climax of the movie. So then the beginning of the new sporophyte generation is here when we form the zygote. Okay, so that's the key. But remember, how many sperm cells were there?
2.
So we still have to figure out what happens to the other sperm cell.
Yes.
So the other sperm cell is also going to join or merge with something in the embryo sac. Do you remember what does the other sperm cell join or merge with in the embryo sac?
The pollen cells.
Good, good. And as you said, it's important to remember there's two polar nuclei in the central cell. Okay. This would be considered another fertilization event. The joining of two cells when they combine their nuclear material is considered fertilization. That's why this whole process is called double fertilization, because there's two fertilization events. Okay. Now, the sperm cell and the two polar nuclei are going to join together to form, and basically in the central cell. And what is that central cell going to turn into eventually?
It becomes the endosperm.
That's the endosperm key. So that's something to really be careful about in our notes. I think earlier we were getting confused and thinking that this was going to be the endosperm. This is something that the instructors definitely want us to be really clear about. This is one of the things that the test will definitely be testing, whether we understand, not confusing the zygote and the endosperm. So this is going to become the endosperm. Okay, so this is a key thing to really highlight. So what's the chromosome number of the sperm cell?
The sperm cell is on the 2.3N.
Now let's make sure we answer the question that I was posing. What is the chromosome number of the sperm cell? Good. And each polar nuclei has a chromosome number of?
Each.
Good. So what would be the new chromosome number of the endosperm?
That would be 300.
That's right. Okay, good. So is the endosperm diploid, triploid, or haploid?
Triploid.
How about the zygote?
Triploid also. Sorry.
The question is, what's the zygote would have what term?
The zygote.
That's right. Diploid, triploid, or haploid.
Diploid.
Yeah, that's once we look at the word diploid means two. Okay. All right, so again, this is some key material. And then what is the fate of the endosperm? Or what's the role of the endosperm? How does the endosperm help this process?
It takes in the nutrients that it needs to form.
That who needs?
Embryo.
Okay, so the endosperm is going to store nutrients for the growing embryo. Okay, so like I said, these are some of the most important material here that we've covered that will certainly be on the test. So the fate of the zygote is to become the embryo. The fate of the endosperm is to store nutrients for the embryo. We also talked about what's the fate of the ovule. What does that develop into?
The ovule is the seed.
Good. Which is what, good, and what the new plant is going to grow out of. And what's the fate of the ovary?
That will have a fruit.
So the hope is that perhaps an animal will eat the fruit and then carry the seeds away and then deposit them in another location so that the plant can grow in another location. Okay, so this is some of that key material that we had covered, that we covered last time. That's important for us to review here. So when I was asking you about the components of the male and the female gametophyte, it looked like we had pretty good recollection of that. But this material, it looked like we were pretty confused about. And this is stuff that will definitely pay off for you if you can review it. This will be on the test. So pretty much everything we just talked about for the last 15 minutes definitely will be on the test and can translate into points for you on the test. But it's confusing. It takes quite a bit of work. The problem is that we don't want to confuse this with some of the other ideas that we talked about. Notice that at one point when we were working on this, you mentioned, for example, say the megaspores. But notice that has no role in this process. That was something that came much earlier in the process. Okay. Okay, good. I wanted to ask you, after our last session, did you have a chance to review the material that we talked about in our last session?
I did. It was just this specific part. I reviewed everything. It's just that I didn't remember just this part of it.
Okay, fair enough. How did you review? So did you review by looking at your notes, or did you review by taking a blank piece of paper and trying to write out the whole process again from memory?
Well, I just got that new sketchbook, and right after I just wrote it on there.
Oh, you tried writing it actually on your screen?
Yeah.
Okay. Oh, you mean today, or
No,
when
yesterday.
Oh, okay,
Well,
that's fine.
after our session.
Yeah, okay, that's fine. If that's what works for you, that's fine. There isn't really any need for you to take your own do your own review on the sketchbook. You only need that when we're doing the Skype sessions. But if you find that helpful, that's fine. So I was going to ask you again. So did you write out the whole fertilization, this whole fertilization thing, on a piece of paper or on the screen to practice it?
Yeah.
Okay.
Yeah, I wrote from when we started to now, and I drew out, the embryo sac and the process it goes through to create the embryo sac and
Okay, good.
the double fertilization process.
And did you get a chance to check that against your notes to make sure it was correct?
Yes.
Okay. But now, so the moral is, it turns out, it looks like that wasn't enough to get you the points on the test, right? Because if you had taken the test today, you wouldn't have gotten too many points for this section. You wouldn't have gotten so that the moral of the story is you need to do that every day.
Yeah.
The moral of the story is you need to try to do that every day, because otherwise, like we talked about last time, like I say, in my tutoring policies, I let people know up front, most people don't benefit from tutoring. And this is you can kind of see how that happens, because it's just so hard to remember all the material. So anyway, I just want to make sure that you consider that going forward. After each tutoring session, it's necessary to keep writing out what we've gone over in the tutoring session every day, if possible. And I'm sure you got plenty of other things to do with your time. But if you want to benefit from the tutoring sessions, you need to keep writing out everything every day, kind of like I was just forcing you to do, because otherwise people quickly forget the material. And that's a shame, because you paid for it, right? It would be a shame to pay for the session and then not get the points for that on the exam. So I just wanted to warn you about that up front. What is pollination?
Pollination is a process where the pollen grains are attaching to the stigma.
Good. And then what is inside the pollen grain?
A tube cell and the generative cell.
Good. So what happens next after the pollen grain sticks to the stigma?
The tube cell turns into the pollen tube, and that allows the generative cell to travel down. Well, the pollen tube ends up traveling down to the ovary, and that what allows the generative cell to move down to the ovary into the ovule, into the embryo sac.
Good. What does the generative cell do?
The generative cell goes through the process of mitosis and splits into 2 sperm cells.
Right. So I think that's right. At first, the generative cell moves into the tube cell or into the pollen tube, but then it creates 2 sperm cells. So it's the sperm cells that complete the journey through the pollen tube. Okay? What do the sperm cells do when they reach the ovary?
They go through the process of double fertilization.
Good. Well, describe what one fertilization involves.
When one sperm, it merges with the egg cell.
Good.
And then it goes through the process that's when it goes through the process of fertilization.
That's fertilization. Good. So when the egg cell merges with the sperm cell, what do you call the product of that merger?
The zygote.
And what's its chromosome number?
2N.
Good. And then the zygote becomes?
The embryo.
Good. And eventually the embryo becomes?
The new plant.
Okay, good. The new sporophyte version of the plant. Good. Now you mentioned there was double fertilization, so what's involved in the other fertilization event?
Wait, should I have been drawing this, or?
That's up to you. You were saying it correctly, and we already have it in my notes on my screen. I'll send you my notes again. So that's up to you. If it helps you to write it down, that's fine. But you already should have a couple copies of that in your notes. Yeah. If it helps you to write it out again, that's fine too. I just want to make sure that it's in your head right now. Yeah. Okay. So what happens in the second fertilization event?
The second sperm cell, it'll merge with the 2 of the polynuclei cells. And they go through the process of fertilization also. And then it'll become the endosperm.
Good. And the chromosome number of the endosperm is?
3N.
Good. And the function of the endosperm is?
It stores nutrients for the embryo.
Okay, so I think we made some good progress on that now. Very good. Okay, so now the key thing is just to keep practicing that every day until the test. Because like I say, those are points in the bank. Those are all things that will definitely be on the exam. But it's so easy to start forgetting all that material. Okay, what are the four basic parts of a flower?
Oh, the carpel, the sepal, stigma, petals.
Let's write those down so we don't get confused. Let's write down the four basic parts of a flower. Of course, there's lots of subparts, but now if you see what I'm saying, we want to write the basic parts, not the subparts. Oops. These are what the textbook calls the four basic parts of the flower. So I was asking you to write down the four major parts of the flower. We don't want to write down the subparts right now, just the four major parts.
Okay. So the carpel, the stamen, the sepal. What was the other? Oh, the petals.
Okay, good. What is the male organ of the flower?
The stamen.
Good. And what's the female organ?
The carpel.
Okay, good. Distinguish between pollination and fertilization.
Pollination is the process where the pollen grains are attaching to the stigma.
Good.
And fertilization is a process where the generative cell of one of the pollen grains become sperm cells and then travel down the pollen tube. And then once those two sperm cells reach the embryo sac or the ovary is when they go through the process of double fertilization.
Good. Now, actually, the last thing you described wasn't really probably considered fertilization. It's kind of the lead up to fertilization. What is the actual fertilization? Fertilization is when the sperm cells merge with new cells.
Okay.
If the test asks you what is fertilization, you can describe the lead up, but you have to make clear that the actual fertilization is when the sperm cells merge with the new cells. So one sperm cell merges with?
The one sperm cell merges with the egg cell to form the zygote.
That's one fertilization event. Good.
Oh, and then should I say
And the other one is
when the other sperm cell merges with the two polar nuclei, and that fertilization creates an endosperm.
Excellent. All right, so that's what they mean when they say to distinguish between pollination and fertilization. Fertilization itself is when the sperm cells merge with the egg and the polar nuclei. Okay. Describe the fate of the ovule and the ovary after double fertilization.
The ovule will become the seed, and the ovary will become the fruit.
Okay, good. Okay, good. So I just wanted to point out to you what we've covered here so far. So I think we've done this objective. We listed the four floral parts in order. I think you could now do number 2, label the sepals, petals, stamen, and carpel. You could do number 3, name the male and female gametophytes. We could do number 4, describe the development of the embryo sac and the fate of its cells. We especially focused on the fate of the egg cell and the polar nuclei. I think we've talked before about how pollen can be transferred between flowers. We just distinguished between pollination and fertilization. This one we haven't talked about. Outline the process of double fertilization. We talked about that today. This we haven't talked about. We haven't gone into the adaptive advantage of double fertilization. This part we just talked about, describing the fate of the ovule and the ovary after double fertilization. We talked about this a little bit. Number 10 we haven't talked about yet. Number 11 we haven't really talked about. Number 12, kind of talked about it a little bit. haven't really talked about 13 or 14. All right, so I just wanted to show you what we have and haven't covered. Maybe, but anyway, so there's a bunch of things here we haven't covered that might be on the test. However, I think what I tried to do was cover what I thought would be most important on the exam. Let's see. But remember that between now and the test, it's important to keep reviewing those objectives that we just talked about, because it is really easy to quickly start forgetting a lot of that material. Let's see here then. Let's actually look at one of these problems from your first exam. Try this problem. Let's try this problem.
All right. During development of male and female gametophytes, nature plant. A.
Okay. Yeah. We talked about that before, how the gametophytes are much smaller than the sporophytes.
Yeah.
Okay, good. What about, say, C? Haploid number is increased to diploid number to form the endosperm. What's true or false about that?
Oh wait, haploid number is increased.
To form the endosperm.
Oh, it becomes, because it doesn't, it becomes a triploid?
Yeah, so for one thing, it does not increase to diploid, it becomes triploid.
Yeah.
Good. And anyway, that's not part of the development of the male and female gametophytes. That's part of fertilization. Okay, so anyway, that was one of the problems from the last exam. I think the first, on that exam, I think you picked C. But now we can see, as we talked about, why A would be the correct answer for that question.
Yes.
All right, what part of the flower protects the floral bud?
The sepal.
Good. Okay, good. I think when you took the previous test, you thought that was the carpel, but this was, now we've learned those types. Good, so that's the sepal. Pollen is released from what structure?
The anther.
Good. This is the landing area for pollen.
Oh, the stigma.
Good. That was part one that I think you got incorrect wrong before, too. The structure provides protection for the egg.
The integument. Oh, wait.
I can see why you might say that, but here's the choices. Which structure provides protection for the egg? Of these choices.
Structure for the egg. Oh, wait. Is it the... Wait, where is it? Yep, sepal. Or the ovule, sorry. It's ovary, ovule, then the egg.
Yeah. So the egg is inside of the ovule. So the ovule is providing protection for it. Okay, good. Here's another problem from the test. Describe just the results of double fertilization in an angiosperm.
The results, the sperm that merges with the egg cell becomes a new plant with the chromosome of 2N.
Good.
And
however, what your instructor would go for is what's the first name that it has immediately after fertilization? The sperm is going to merge with the egg cell to form a new cell whose name is...
Zygote.
That's what they'd be going for on this question. Okay. Good. You could say that later it becomes the new embryo and plant, but the key thing is they're going for zygote. Okay. So to continue, what other are the results of double fertilization?
When the sperm cell merges with the two polynuclear cells to create an endosperm.
Good. All right. Good. So that's another important problem. Okay. So it looks like the first time you did this, you said the results of double fertilization would be a 3N zygote. What's wrong with that?
The zygote is the zygote does not have Wait, why did I say that? Zygote's only 2N.
That's right. And also, we should have also said the endosperm. Good.
Yeah.
Okay, good. So this is just to review that. Material. List 3 methods used by plants to disperse their pollen. 3 methods used by plants to disperse their pollen.
Insects, animals, and wind.
Say that one more time.
Insects, animals, and wind.
If you think about that, that's kind of repetitive because insects are animals.
Oh, okay. Insects, water.
There you go. All right, that one you got right when you took it the first time. But that's something else that's likely to come up again on the exam. Okay, now I would expect those were those were the key problems on this chapter from the first exam. I would expect that if when they come up on the next exam, there'll be more of the short answer essays about this chapter. Whereas here it was mainly more really short answers there. Okay, so those would be good things to review. So I think we made some progress on that. Well, the question I was asking you is, what is a angiosperm?
Oh, the angiosperm is basically the plant.
Angiosperms are plants. Good. What type of plant? What's the difference between angiosperms and other plants?
Angiosperms can, well, they contain ovaries, ovules, and then the embryo sac.
Okay, that's right. They do have all those things. We haven't quite described, though, what the basic definition of an angiosperm is, though. Angiosperms are flowering plants.
Okay.
Okay. What's an angiosperm?
Flowering plants.
Very good. What are the male and female gametophytes for angiosperms? Just their names.
Okay, the male is microsporophyte, and the female is megasporophyte.
Okay, so what should I write here?
For male gametophyte, microsporophyte.
Like this?
Yes.
And how about for female gametophyte?
Megasporophyte.
Okay, so those are mistakes. So these cells are, so those are not the correct names here. The male gametophyte
is the pollen grain. I apologize.
Okay, that's okay. But it's important to be clear about that. That's one of the objectives from the reproduction chapter. That's pretty sure to be on the test. So let's make sure we're clear about that. So the male gametophyte is the pollen grain. Okay, good. And then this name was mistaken. What's the name of the female gametophyte?
The embryo sac.
That's right. Okay, so those are names that are very likely to be tested. Okay, so what's an angiosperm?
An angiosperm is a flowering plant.
In angiosperms, what's the male gametophyte?
Pollen grain.
Good. And what's the female gametophyte?
The embryo sac.
Okay, good. So, what is pollination?
Pollination is when the pollen grains released from the anther. Well, it's when the pollen grains are attaching to the stigma.
That's right, when they move from the anther to the stigma. Good, that's a good answer. So after pollination, what happens?
After pollination,
let's try to describe step by step what would be the steps continuing after the pollen grain sticks to the stigma. What would be step by step the steps that happen next?
Okay, well, the tube cell inside the pollen grain would become the pollen tube and begin to travel down the style into the ovary, and then the generative cell will become two sperm cells, and those will travel down the pollen tube into the ovary, into the ovule, into the egg, into the embryo sac.
Good. Okay, excellent. That sounds like a very good description so far. That's very good. Okay, so far so good. What next?
When the two sperm cells enter the embryo sac, they go through double fertilization, and when one of the sperm cells will merge with the egg cell, and then both of them have the chromosome number of N, go through fertilization to form a zygote, and after they form the zygote, they form the embryo, and then after the embryo, they form the new plant, the mature plant, and then the chromosome number of the new plant will be 2N.
What's the chromosome number of the zygote?
2N.
Yeah, that's when the 2N sporophyte generation begins. Okay, that sounds like a very good description. That's a very good description of that fertilization process. So what else is involved in double fertilization besides the sperm cell merging with the egg cell?
The other sperm cell will merge with the two polar nuclei, and then during that fertilization, it becomes a triploid, and that becomes the endosperm, which has the chromosome number of 3N, and the endosperm is used in order to store nutrients for the new plant. Endosperm is in the seed.
Okay, that's also quite good. Very good. Distinguish between pollination and fertilization.
Pollination is the process basically before fertilization happens. Pollination is what allows fertilization to take place.
Good. Let's just be more specific about what exactly happens. And now we don't have to go through all the steps, but what's the basic definition of pollination? What's the key event in pollination?
Yeah. Pollination is when the pollen grain when it attaches to the stigma.
From the anther. Okay, good. It's released from the anther and reaches the stigma. And then what's the key event or events in fertilization?
And fertilization is when the generative cell from the pollen grain become the two sperm cells, and they will travel down the pollen tube.
Good. Now, actually, that would be considered the preliminaries to fertilization. What are the actual two key events, the two key fertilization events?
The two sperm cells merging with the egg cell, and then one of the sperm cells merging with the egg cell, and the other sperm cell merging with the two pollen nuclei cells.
Okay, that's good. So these processes right here, those are fertilizations. What you were describing before was accurate about how the generative cells form sperm cells and then they move down the pollen tube. That's true, but those are not considered fertilization. Those are the preparation for fertilization. Okay, very good. So that's a good review of those topics. So we've said that the zygote eventually becomes the embryo, which becomes the new plant. Now, the this fertilization gives rise to something that becomes the endosperm, whose function is to store nutrients. What's the fate of the ovule?
The ovule will become the seed.
And what's the fate of the ovary?
The ovary will become the fruit.
Good. Okay. All right, now let's talk about something we haven't had a chance to talk about before. One of the objectives for this chapter is explain the adaptive advantage of double fertilization. What is the advantage from an evolutionary standpoint of double fertilization? So we can talk about that together. So the key thing is, what is the function of the endosperm?
It basically provides nutrients for the plant to grow.
Yeah, it stores nutrients. So would you agree that making endosperms is expensive for the plant?
Yes.
Because nutrients are expensive. So would you agree that you don't want to make an endosperm that's not going to be needed?
Yes.
Okay, good. So you only want to make the endosperm if you actually have a growing if you actually have a growing embryo. Well, notice how double fertilization ensures that. Notice that generally speaking here, the endosperm is not going to be created unless the endosperm is not going to be created here unless the egg has also been fertilized. Notice they're basically fertilized together. So that's the adaptive advantage here. The advantage of having the endosperm also depend on fertilization is generally the endosperm won't be created unless the sperm cells have made it to the ovary. Well, if the sperm cells made it to the ovary, then we can be confident that the zygote is going to be fertilized as well. So the adaptive advantage of this is that it ensures that the endosperm is not created unless it's needed for the zygote. Does that make any sense?
Yes.
Okay, good. Okay. Sounds like maybe you're taking some notes. That's good. Tell me when you're ready to keep going.
I'm ready.
Okay. Okay, so let's say there was an essay question that said, explain the adaptive advantage of double fertilization in angiosperms. How would you outline what your answer would be?
I'm sorry. I have to raise the volume. Your mic is like, I can hear you now. Okay, the adaptive advantage would be that because instead of creating endosperms that the plant may not need, the plant will know, it will at the same time it will fertilize to the sperm cells and two polar nuclei will create an endosperm when it knows that the sperm cell and the egg cell are creating the and fertilizing the zygote to the embryo to the new plant. It allows them not to create wasted endosperm.
Good. And why are endosperms expensive?
Because because nutrients are expensive.
That's right. I don't remember whether you mentioned that or not, but that would be an important thing to mention in the answer. Okay, good. Okay, good. So that sounds like the right basic ideas. All right, so that's one of the objectives from the chapter we didn't get to before, but I think there's a good chance that would be tested, so that's a good thing to add to your notes. Okay, so moving on, what happens to the zygote? As the zygote grows, what's it called as it's growing? Well, we know it's going to be called the embryo. Let's actually put in a little bit more detail that you're probably going to be tested on here. How many cells are there in a zygote? How many cells in a zygote? When the zygote is, or maybe this will be clearer, when the zygote is first formed, how many cells does it have?
I'm not sure.
Okay. Well, remember, how is the zygote formed? What's the process by which the zygote, what's it formed out of?
Oh, fertilization. Oh, would it have one cell?
That's right, because these two cells have merged, right?
Yeah.
We took two cells that each have N chromosomes, and we combined them into one cell with two N chromosomes. So by definition, the zygote is one cell. That's something we should have made clearer before. That's the difference between the zygote and the embryo. So the zygote is one cell with two N chromosomes. Again, that's the whole point of fertilization. That's the definition, or that's what's happening in this fertilization. We take two cells with N chromosomes each and combine them into one cell with two N chromosomes. So then how does the zygote grow? Is it going to grow through a process of meiosis or mitosis?
Mitosis?
We've already finished with the meiosis. That was what we used way back in the process to start the creation of the female gametophytes. Normal growth comes from mitosis. Normal growth comes from mitosis. So first of all, we could have one round of mitosis. So let's say we have one cell, which is the zygote, and it goes through one round of mitosis. How many cells would there be after that one round of mitosis?
There are two more cells.
That's right, because remember we reviewed at the first session how mitosis works. Mitosis takes one cell and changes it into two cells. So now we have two cells. What's the chromosome number of each cell?
2N.
Because remember, mitosis, as we reviewed earlier, always maintains the same chromosome number.
Yes.
All right. Now, I think you're likely to be tested on the names of these two cells. Have you happened to learn this yet? What are the two daughter cells of the zygote?
Wait.
So these two daughter cells of the zygote have names. Have you happened to learn those names yet?
I don't think we have.
Okay.
Wait.
Oh, well, I was just asking whether you yourself happen to remember those from class. So
No.
these were two names that were covered in the notes. Let's talk about that. So one of these cells is called the terminal cell.
Mm-hmm.
So what we're doing here is we're just tracing out the growth of the zygote in a little bit more detail that I think is likely to be tested. So the zygote turns into the terminal cell and what's called the basal cell.
All right.
Okay. After the zygote goes through one round of mitosis, what are the two daughter cells, the names of the two daughter cells?
The terminal cell and the basal cell.
That's right. And now we need to know the fates of these cells. So the terminal cell is going to become the embryo, or the main part of the embryo. And the basal cell is going to become what's called the suspensor. The function of the suspensor is to anchor the embryo to the parent plant. So remember, this whole process is still taking place inside the ovary of the parent plant. So the suspensor is a kind of connection to the parent plant. Nutrients can flow through it. It's kind of like the umbilical cord. Okay, so the suspensor here is the anchors the embryo to the parent plant. These are some new terms that I think are likely to be tested. Okay. So when the zygote divides, it turns into two daughter cells. What are the names of the two daughter cells?
The embryo. Well, it's the terminal cell and then the basal cell.
That's right. It becomes the terminal cell and the basal cell. What's the fate of the basal cell? What happens to the basal cell?
The basal cell will become the anchor.
Good. What's the name of the structure it forms?
The suspensor.
Suspensor. Very good. And it anchors the embryo to the parent. Good. And what's the fate of the terminal cell?
The terminal cell will become the embryo.
That's right. It becomes the embryo. All of these obviously are still 2N. And then eventually the embryo is going to grow into the new plant, the new sporophyte plant. Okay, so those are some extra details that I think are likely to come up as we go through this. Okay, so very good. Now, continuing then, where the growing embryo is going to need nutrients. Where are the nutrients stored for the growing embryo?
In the endosperm?
In the endosperm. Okay, good. However, there's another really important structure that also in the seed that also stores nutrients that we haven't talked about yet, but is definitely important on the test. There's another very important structure that stores nutrients. The other important structure that stores nutrients is called, excuse me, cotyledons.
Mm-hmm.
Cotyledons store nutrients for the growing embryo. So notice that they're quite similar to the endosperm. Now, the cotyledons are seed leaves. That is, they're basically little leaves that exist inside the seed. They're tiny little leaves inside of the seed. Okay. Another term that comes up, well, let's make sure we got that down. What's the function of the cotyledons?
It's also what stores the nutrients.
Like the endosperm. That's right. And they're seed leaves. Okay, good. But they're not like a regular leaf. The function of a regular leaf is photosynthesis, and these are not doing photosynthesis. At least yeah. All right. So now some more terms that come up a lot. There's monocot. Do you know what this means?
Oh, a monocot is. It's when I believe no.
So let's talk about
monocot,
that. So do you remember what does the prefix mono mean?
One.
That's right. And we need to contrast those with eudicots. Do you know what does di mean?
Two.
Yeah. Well, take a guess. What do monocots have one of? So these are two different types of plants. These are the two major types of angiosperms. The two major types of angiosperms are monocots and eudicots. Monocots have one what? Could you take a guess from the name? What do they have one of?
Nuclei?
They actually have one cotyledon. And then what would be a eudicot?
They would have two cotyledons.
That's right. Okay. So monocots are, so these are just two types of plants.
Yes.
These are a type of plant, a type of angiosperm with one cotyledon in each seed. So what's a eudicot?
Type of plant with two cotyledons in each seed.
Okay, good. There's certainly more details we could get into, but I just want to cover the most important points that are most likely to be tested. Okay, so, all right, very good. So, good. What's a cotyledon? Let's see. What is a cotyledon? When you're ready.
I'm sorry.
Yeah, take your time.
Could you? Hello?
Hi, can you hear me?
Yes.
Okay. So I was asking, what is a cotyledon?
Cotyledon is also, it's along with the endosperm, it also stores nutrients.
Good. And it's also good to know what type of structure is it. Those are what are considered, those are the seed leaves. Okay, good. And what's a monocot?
Monocot, it contains one cotyledon.
It's a type of plant whose seeds have one cotyledon. Good. Why is monocot a logical name for that type of plant?
Because it only has one cotyledon.
Yeah, mono means one, and the cot refers to the cotyledon. And what's a eudicot?
Eudicot is a type of plant that contains two cotyledons.
In each seed. Good. So why is eudicot a logical name for that type of plant?
Because it has di in it, which means two.
And cot, which means cotyledon. Good. So I'm just pointing that out to make it easier to remember what those terms mean. Okay, very good. So let's see. We know that in double fertilization, an egg cell is going to be fertilized by a sperm cell. What do you call the product? What do you get when an egg cell fertilizes, when a sperm merges with an egg? When sperm merges with egg, what do you call the product of that?
A plant.
What do you call the very first product?
Oh, the zygote.
The zygote. How many cells are in the zygote?
Just two. Or one, sorry.
Yeah, the zygote has one cell after fertilization. 2N cell. Good. Then it goes through one round of mitosis to make two daughter cells. What are the names of the daughter cells?
The terminal cell and the basal cell.
That's right. Good. And then what are the fates? What's the fate of the basal cell?
The basal cell and the terminal cell. The basal cell will become the I forgot the word. It becomes the anchor, the suspender.
That's close. Good. That's right. So it's the suspensor.
Okay.
Good. And what's the fate of the terminal cell? When you're ready.
The terminal cell itself will become the embryo.
That's right. And what's the fate of the basal cell?
It'll become the suspensor.
Very good. Okay, that's right. And again, the terminal cell becomes
the terminal cell will become this is. Wait, did we just talk about this?
I'm sorry. I'm just trying to review so that they're clear.
Oh,
So
okay.
yeah. So we did just talk about it. What's the terminal cell become?
The terminal cell becomes the embryo.
Okay. So the terminal cell will continue dividing by mitosis to become a pro-embryo and then an embryo. However, you want to keep in mind that pretty soon this dividing embryo will become dormant.
Okay.
Because remember, all of this is still happening inside the parent plant. There's no point having the seedling grow inside the parent plant. So after the embryo has divided some more and becomes somewhat larger, eventually it's going to become dormant. Mm-hmm. Okay, so then what happens next after the embryo has grown as much as it's going to grow at this stage and it's become dormant? What's kind of the next stage of the process?
Does it become the plant?
Well, remember, it's dormant, so it's not becoming the plant right away. What events are going to have to happen? What events are going to have to happen before the embryo continues its growth?
Germination.
That's right. But what events are going to have to happen? So where is the embryo right now at this stage?
It's still in the ovary or in the ovule?
Yeah, in the ovule, in the ovary. And where's the ovary?
In the plant itself?
Yeah, in the parent plant. Good.
Yes.
So right now it's still in the parent plant. Mm-hmm. Okay. So what is the next event that has to happen now? What will be the next step in the process?
Did we discuss this?
Yeah, that's right. And it's also a logical step.
Oh, the seed is taken by the animal?
Okay, yeah, that's right. Why would the animal take the seed? What's going to attract the animal to take the seed?
Would it be the colors of the petals?
Now, the petals are really involved in attracting pollinators.
Yeah, or basically would it be the nutrients that the animal would need from the plant?
Well, if the animal ate, we don't want the animal to eat the whole plant because that would just kill the plant. So the specific thing what is going to attract the animal to carry the seed, what we haven't mentioned here is the fruit.
Okay.
All right, that's the function of the fruit. All right, so the embryo becomes dormant in the seed. Then the next stage is that the fruit is carried to a new location. So this is something we've talked about somewhat before, but we haven't really clarified it. So now is an important time to do that. After the embryo becomes dormant in the seed, well, I guess that actually, so now the fruit is going to be carried to a new location. Okay.
And then eliminated by the animal.
That's right. So what could carry the fruit to a new location? The textbook mentions two major things that could carry the fruit to a new location.
Wind and insects?
By wind or animals. When you mentioned insects, you were probably thinking of pollination. Notice it's easy to get these different stages confused. Insects are really important for pollination in many cases. However, insects are not particularly important for spreading seeds, right? Because most insects are not big enough to swallow a whole fruit, right? True. Most insects are not big enough to swallow a whole fruit. So most of these fruits would be carried by other types of animals, like maybe birds or other animals that can reach up and eat the fruits. Okay, so the fruit is carried to a new location. Some fruits might have wing structures that can allow them to be carried by the wind to a new location. Other fruits could be swallowed by animals, and then the animals can carry them to a new location, and then when they eliminate the waste, the seeds are expelled into the soil. Or some fruits also have little burrs that can stick into the fur of animals, and that also would carry them to a new location. All right, so one way or the other, the fruit is going to be carried to a new location by wind or animals. If the fruit is going to be eaten, this would be a good time for the fruit to ripen. right?
Okay.
This would now be a good time for the fruit to ripen so it could be eaten. So this gives us a good chance to connect this with another chapter. What hormone would be released at this stage to make the fruit ripen?
Ethylene.
That's right. Okay, so that's a connection between what we're talking about now and one of the other chapters. So this would be a good point for the plant to release ethylene to cause the fruit to ripen. Okay. Okay, so when you're ready, we've been talking about that the embryo grows in the seed for a while inside the parent plant, but eventually the embryo is going to become dormant in the seed in the parent plant. So what's the next thing that has to happen after the embryo becomes dormant in the seed in the parent plant? What's the next step in the process? The question was, what's the next step after this step? After the embryo becomes dormant in the seed while it's still in the parent plant, what has to be the next step in the process? We were talking about how at first the embryo grows in the seed inside the parent plant, but eventually that process stops and the embryo stops growing and becomes dormant. And I was asking you what happens next. What is the next step in this process? After the embryo has become dormant in the seed while it's still in the parent plant, what happens next?
Doesn't the animal eat the what becomes the fruit, and then that's how the seeds are transported?
Okay, yes. So the next step is that the fruit is going to have to be transported by animals or by wind to another location. Okay, good. And then one way or another, the seed is going to be deposited in the soil. So the fruit is carried to a new location by wind or animals. So at this point, now the seed is in a new location. But initially, it's still dormant. Okay. And then remember, eventually, when the seed senses that conditions are favorable, it'll start growing again in the soil in this new location. What's the name of that new process when the embryo in the seed now resumes its growth and starts growing into a plant in the new location? What's the name of that process?
Germination.
Exactly. Good. When the seed senses that conditions are favorable, we have germination. Okay, good. And let's talk about that in a little bit more detail again. We talked about this in the previous session. Do you remember what is generally the first event that starts or begins germination, or an event that occurs at the very beginning of germination? That might seem like a weird question, so let's review that together. So the first thing that happens is what's called imbibition. We talked about that last time. What is imbibition?
Imbibition is basically when water enters the seed.
Good. And that leads to germination. The embryo resumes its growth and becomes a seedling, and eventually the new plant, and eventually a new mature plant. Now, okay, good. So, let's see. So, to review these steps, the sperm cell fertilizes the egg cell, and what do we call the product of that fertilization?
Zygote.
Yes, that's a single cell. What happens next with the zygote?
The zygote will become the embryo.
Eventually, yes, but we've now broken it down into smaller steps. So what's the very next step for the zygote?
Well, the two cells will form.
Good. What are the names of the two cells?
Terminal cell and the basal cell.
Very good. What's the fate of the basal cell?
The basal cell will become the I don't want to say the same word. The suspensor.
Suspensor. That's right. Which anchors the new plant
embryo
to the parent plant. Good. And what's going to happen to the terminal cell? What's its fate?
That will become the embryo itself.
That's right. That will become the embryo. So the terminal cell continues dividing and becomes a growing pro-embryo and then a regular embryo. But that process doesn't continue indefinitely. Eventually, while the embryo is still inside the parent plant, it'll become dormant. And then what happens next to continue the process after the embryo has become dormant inside the parent plant?
After the embryo becomes dormant, an animal will eat the fruit and then eliminate it into a new location where it can start the process again.
Very good. That's right. What's the other mode by which fruit can be spread besides animals?
Wind.
Good. So there's likely to be a test question. Name two methods by which fruit is spread. So those would be animals and wind. Good. Okay, good. So the fruit is carried to a new location by wind or animals. Then what?
And then the animals will eliminate the seed into the new location, and then basically it will go through imbibition.
That's right. What is imbibition?
It's when the water enters the seed to allow it to grow.
Yeah. So that should occur when conditions are favorable because there's plenty of water. Good. And then imbibition is the first step of what process?
Germination.
That's right. So it's important on the test not to confuse pollination, fertilization, and germination. Those are all three different things. So imbibition is the first step of the germination process. Okay, very good. Let's put in a little bit more detail. So after imbibition occurs, there's going to be a hormone released that's going to stimulate germination. This is something we talked about last time. What is the name of the hormone that stimulates germination to begin?
Gibberellin.
Gibberellin. Gibberellin is released. And that will stimulate germination. So that's a connection between these chapters. So the gibberellin is released as a result of the water entering the seed, and then that stimulates germination to continue. Okay, very good. Good. So when is germination going to be ended? Germination is going to end in response to signals from the environment. For example, if there's enough water, or we talked last time about how if a period of cold is coming to an end, that's a signal to the seed that the winter is ending and the spring is starting. So one of the signals for germination to begin might be that it seems like winter is ending and spring is starting. So there's a variety of signals that can signal germination to begin. One thing that's worth keeping in mind is what happens if conditions are not good for germination. If conditions are not good for germination, the seed will just stay dormant. Some seeds can stay dormant for decades. Some seeds can stay dormant for a really long time, decades or more. So that's an interesting aspect of this idea. Okay, very good. One moment. Okay. What is a monocot?
A monocot is a type of plant with only one cotyledon.
Good. And what's the other type of plant?
Dicot.
Good. That's logical. Actually, the name for the name for that group is eudicot.
Oh, eudicot.
So dicot would be logical, but actually the main group of dicots are called eudicots. So that's an important name to know. Good. Okay, so the process that we've been discussing here, have we been talking about sexual or asexual reproduction in plants?
Asexual.
So did you say that process was asexual?
Yes.
Any particular reason why you use that term?
Well, I mean, I guess we've been using.
So what I mean is this whole process we've been talking about today, is double fertilization part of sexual or asexual reproduction?
I guess sexual because you're both. Well, not I guess it's sexual because you're using a sperm and an egg.
Okay, that's right. So sexual reproduction, it means there's two parents. Who are the two parents here? Well, technically the two parents are the male gametophyte and the female gametophyte. Those are the two parents. The two parents are the male and the female gametophyte. They're producing the gametes that come together here in fertilization. So what we've been talking about is sexual reproduction, where two parents are producing the offspring. So everything we've talked about so far was sexual reproduction. So flowering plants can definitely undergo sexual reproduction. Can flowering plants use asexual reproduction?
I don't think so.
That's a good thing for us to talk about. Actually, it turns out that flowering plants can often also do asexual reproduction. Not always, but asexual reproduction is often also possible for angiosperms. So there's a variety of different methods of asexual reproduction for angiosperms. One that your textbook especially focuses on is just fragmentation. And fragmentation just means that part of the plant can separate from the parent and just grow into a brand new plant. So, for example, a part of the stem of the plant might just fall off into the soil, and it might just grow into a brand new plant. This is something that people kind of already know from their everyday life. You often know that you can kind of cut, if you have a house plant, you can often cut the house plant in two and plant the two halves, and they grow into two new plants, right? So you can use that. People can use that, but plants can also use that for reproduction. A part of the plant can simply fall off, like a piece of the stem can fall off, and it can just simply grow itself into a whole new plant. Obviously, this is not something that most animals can do. Most animals can only do sexual reproduction. So the textbook used a good analogy. The analogy the textbook uses is it would be if somebody cut off your finger, they couldn't grow a new version of you from the finger. Right?
Yeah.
So for human beings, there's no such thing as this type of asexual reproduction. You can't just cut off someone's finger and grow a new person from the finger. But that actually really is what many plants can do. Many plants can just have a piece of themselves fall off and then grow into a whole new normal plant. Now, is asexual reproduction going to give you offspring that are different or identical to the parent?
Oh, identical?
That's something that's likely to be tested. Asexual reproduction gives us offspring that are identical to the parent. So it proceeds purely by mitosis. Does sexual reproduction give us offspring that are the same or different from the parent?
Different from the parent.
So that's an important difference between these. By the way, one of the names for the offspring by asexual reproduction is clones. A clone is just an identical copy of the parent. A clone is just an offspring of the parent that has the same genes as the parent. So clones have the identical genes to the parent. So fragmentation produces a clone. Okay, so that's it's important to realize that even though we've been focusing on sexual reproduction, plants can also do asexual reproduction, for example, by fragmentation that produces clones. So remember, what is the purpose of a flower? A flower is the reproductive organ of an angiosperm, but is the flower used for sexual or asexual reproduction?
Sexual reproduction.
Yeah, the flower was part of the sexual reproduction, double fertilization process we've been talking about. It's not necessary. It's not a necessary element of asexual reproduction. Okay, very good. Okay, so what is the name of the male gametophyte?
Pollen grain.
Good. And what's the name of the female gametophyte?
Embryo sac.
Good. Let's review one more time the process of the creation of the embryo sac. One of your learning objectives is describe the development of an embryo sac. I think there's a really good chance you'll see an essay question like that. So let's move to your screen and review that one more time. Remember that in our first session, we described step by step the entire process of the development of an embryo sac. I think there's a really good chance that'll be tested. So you remember in our very first session, we described in detail the formation of the step-by-step process that involved in the development of the embryo sac. Like I say, I think that's likely to be tested. So let's try writing that out again, like we did in the first session. Try to write out just outline what the key steps are in the development of that embryo sac.
Okay, so first, the female gametophyte, the megasporophyte will. Hello?
I'm here. Yeah.
Oh, okay. The
just go ahead and try diagramming it out. That's good.
With chromosome number of 2N. We'll go. Oops. We'll go through meiosis. And then, as that happens, it'll create 4 new daughter cells called megaspores. And they contain the new. They will die, and then that. Okay. The one megaspore.
So 3 of them will die. Good.
I'm gonna go through 3 rounds of mitosis, and then those 3 rounds of mitosis will give you 8 nuclei, and then those nuclei will begin to form their own membranes.
Good.
And then there will be 2 central nuclei, which are called polar nuclei. Okay.
Okay. So what is the name of the female gametophyte?
The embryo sac.
Okay, good. I wanted to review this because when we went over this at the beginning of the session, at first, when I asked you for the embryo sac, you had mentioned the megasporophyte. So I wanted to make sure that we clarified that. So now, but this was actually a very good outline. This is a very good way to outline the key ideas. So one thing we should correct here is it's not actually called the megasporophyte, it's the megasporocyte.
Oh.
With a C. You were probably confusing that with the gametophytes and the sporophytes. That's where the phytes come in. So it's a lot of stuff to keep track of. Okay, very good. So the key thing is the megasporocyte the female gametophyte?
Yes.
Is the megasporocyte the female gametophyte?
Is the embryo sac is?
Yeah. So it's not. Instead, it's kind of the parent of the female gametophyte. It gives rise to it. But the megasporocyte is still part of the sporophyte generation. You can tell because it's 2N. Whereas the new nucleus is here. As you said, the megaspore is the beginning of the gametophyte because it has N. Okay, good. We also went over the creation of the male gametophyte. What's the name of the male gametophyte?
Microsporocyte.
The question is, what is the name of the male gametophyte?
I keep getting these mixed up. It's pollen grain.
That's right, yeah. So it looks like there's a tendency to confuse those. The microsporocyte is kind of the start of the process that gives rise to the pollen grain, but it's not the same thing as the pollen grain. They don't have the same chromosome numbers. Okay, well, anyway, to save time, I'm not going to review that with you right now, but that would be a good thing to review as well. Anyway, so I think you definitely have the basic idea here, but it's easy for us to get a little confused. Okay, but overall, that was actually a very good process of diagramming the process of the creation of the embryo sac. I think that would be a good basis for an essay answer there. So that's some good progress and a good recollection of that topic. Let's move back to my screen.
He said this is one of the things that's going to be on chapter 30. This is the one essay question that he's going to put.
He said there would only be one essay question about it?
He's going to put like multiple choice questions about chapter 38, but this is going to be the essay question.
Oh, okay. Well, that's good to know. Okay. You tell me. So what's happening?
This is basically when the water will enter the endosperm, and that's...
So what's the name of this process right here? The water is entering?
Imbibition.
Imbibition, sorry. That's right. And then what's happening here? Do you know what the GA stands for?
No.
So this is a hormone that's released to begin germination. So what must the GA stand for? What's the hormone that is released to begin germination?
Gibberellic acid.
That's right. Actually, it's not gibberellic acid. It's gibberellin. I don't know why it's called GA. It's not gibberellic acid. You might be confusing it with abscisic acid. So yeah. I'm not sure why they use the abbreviation of GA for that. It doesn't seem very logical. But anyway, this stands for gibberellin. So at this point, the gibberellin here is being released in response to the water. And that's going to begin the process of germination. So what happens as a result of that? That causes this outer layer of the seed to release amylase.
And the enzyme.
Excuse me?
That's in the enzyme?
The amylase is an enzyme.
Okay.
Yeah, amylase is an enzyme. And notice that the amylase is being released into this portion, which has labeled as the endosperm. So what's the function of the endosperm?
To store nutrients.
Right, but we don't want to store them anymore, right? Now's finally the time to use up the nutrients. So that's the purpose of the amylase. The amylase now will start breaking down the nutrients. It breaks them down into what? Well, sugar. Sugar is one of the key nutrients that the plant can use. So the amylase is breaking down the nutrients into sugar from the endosperm, and then you can see those nutrients are moving over here into the growing seedling.
Okay.
So that basically is the basic interpretation of what's happening in this picture.
Okay. All right.
Okay. So to review what's happening in this picture. What's the name of the process that's happening in this first picture?
Imbibition.
Yeah, and then what's happening in this picture?
Sorry, could you repeat the question?
You said that in this picture we had imbibition. Then
Yes.
what's happening in this picture?
It's the hormone gibberellin is being activated.
That's right. And then over here,
The enzyme amylase is being released from the aileron, I guess. It's from
yeah, from the, I don't know how to pronounce that, but from the aileron, which is this outer layer of the endosperm. What was the signal to release the amylase? What told the amylase to get released?
Oh, the gibberellin.
Yeah. So the water is the signal to release the gibberellin. The gibberellin is the signal to release the amylase. And then what does the amylase do?
The amylase will become sugar.
Say that one more time.
The sugar. Hello?
Yeah, I'm here. Can you hear me?
Yes.
Okay. So just repeat one more time. What does the amylase do?
The amylase will become sugar, and that's what's going to allow the plant to begin to grow.
Okay, so let's talk about that a little bit more. The amylase is an enzyme, so it's not a sugar. Enzymes are little machines, so the amylase doesn't become sugar, it makes sugar. What does it make sugar out of? It makes sugar out of the endosperm. Remember that the endosperm is storing nutrients. The endosperm is storing nutrients in large molecules. And then the amylase breaks those large molecules into smaller sugars that can be used for nutrients. So the amylase is not a sugar itself, it's an enzyme that converts the endosperm into sugar. The amylase converts the endosperm into sugar.
Okay.
And then what happens to the sugar?
The sugar will go into the plant, and that's what's going to allow the plant to grow.
That's right.
Yeah.
Notice, what is this yellow structure here? You can see this yellow structure is the growing embryo. So notice the sugar is moving here to the growing embryo. You can see that the embryo has roots that are extending down, and it also has a shoot that's extending up. Okay. Well, since your instructor was kind enough to tell you that this would be the essay question, let's just make sure one more time that we're prepared for that. So what's happening in this picture?
In this picture, basically, imbibition is happening.
Good. And then what happens in this picture?
The endosperm is getting the signal that gibberellic acid needs to be released from the aileron.
Yeah. What is the signal that causes the gibberellin to be released?
Water entering the endosperm.
Yeah, I think we didn't quite put that right. If you look at the picture, I think you said gibberellin is released from the aileron. But actually, if you can see from the picture, where does the gibberellin actually come from?
The embryo.
It's actually coming from the embryo. What tells the embryo to release the gibberellin?
The water entering the embryo.
The water. But then where does the gibberellin go?
It goes to the aileron?
Yeah, that's right. And what does it cause the aileron? You can see that from the arrows, right?
Yeah.
And then what does it cause the aileron, which is the aileron is the outer layer of the endosperm. Remember that this section here is the endosperm. What does the gibberellin cause the aileron to do?
To release the enzyme amylase.
That's right. That's good to mention. This is an enzyme. What does the amylase do?
And then the amylase will break down the nutrients of the endosperm, or the molecules of the endosperm, to produce sugar.
Good.
And that, okay.
And the sugar?
And the sugar will go into the embryo of the plant, and then that will allow the shoot to rise.
By providing nutrients. Okay, very good. Okay, so the aileron you can see is the outer layer of this endosperm. This yellow section is the embryo. By the way, notice you can see from the picture, what's this? It's the cotyledon. We've already talked about that, right? So this is the cotyledon. In this particular type of plant, this barley seed, there's a special name for the cotyledon, which is the scutellum. But anyway, this is the cotyledon. It's not playing too big of a role in this particular picture. The radicle is the part of the embryo that grows into the root. You can see that from the picture. The radicle is the part. But that probably would not be necessary to mention in your essay answer. So yeah, I just wanted to mention those. But actually, in your essay answer, the cotyledon and the radicle are not really playing a very big role. The key things are water, gibberellin, alerone, amylase, sugar, and the growing embryo. And these are all, this is what's called, how would you know to do this? This is the mobilization of nutrients during germination. This is called mobilization of nutrients. Remember, what's the function of the endosperm?
To store nutrients.
But how does the endosperm know when to release the nutrients? Well, this is the process that tells it to release the nutrients. Okay.
So that would be a question, like how would the endosperm, I guess, how would nutrients be moved throughout the endosperm?
How would nutrients be mobilized during germination?
Okay.
That would be a good, how are nutrients mobilized during germination would be a typical way to phrase that.
All right.
Okay, does that make sense?
Mm-hmm.
Okay, so yeah, since you know that's what the essay is going to be about, this would be a good thing to go back and review. Just wanted to point out that we just went over here is this objective, the mobilization of nutrients during germination. And now we've also gone over a few extra things here. So we went over, we've already talked about the embryo sac. What's the adaptive advantage of double fertilization in angiosperms?
The adaptive advantage would be once the sperm cell is merging with the two polar nuclei, once they know that the other sperm cell going through fertilization with the other egg cell, they know that they both need to go through the process of fertilization, so they don't produce wasted endosperm because the nutrients are valuable. Okay,
excellent. That's good. That might be a little bit, you might be personifying a little bit much, because of course they don't know it. The point is that because the fertilization is double, the endosperm is not going to be fertilized unless, so the point is you don't want the endosperm to develop unless the egg has been fertilized. And that's what double fertilization ensures. The endosperm will only develop if the egg is being fertilized as well. That's basically what you said. That's basically what you said. Okay, good. Another objective, note where major nutrients are stored as the embryo develops. So what are the two main structures we talked about that store nutrients for the embryo? We discussed two main types of structures that store nutrients. What were those?
The endosperm and the scutellum, the cotyledon.
Yeah, the general name is the cotyledon. The scutellum is a name just for one particular type of plant. Okay, good. It's a type, I think, of monocot. Describe the mobilization of nutrients during germination. Well, that's what we were just talking about. It starts with imbibition, release of gibberellin, and then the amylase that turns the endosperm into sugar. What are the two main methods for dispersal of seeds?
Wind and animals.
Okay, good. We talked about how the egg cell turns into a zygote. And what happens? What's the next thing that happens to the zygote? What does the zygote become?
Well, the zygote will become two separate daughter cells, the terminal cell and the basal cell.
So that's this part of the picture.
Yes.
And then what's the fate of the basal cell?
The basal cell will become the suspensor.
So you can see that happening in this picture. Good. And you can see the terminal cell becomes the pro-embryo, which turns into the regular embryo. The cotyledons being formed, remember those are the seed leaves. And you can see how the suspensor is anchoring the embryo to the parent plant at this point. But I think we made some good progress on that. Obviously, it's important to keep reviewing that before the test comes around. Well, what is an angiosperm?
An angiosperm is a flowering plant.
That's right. So what is the angiosperm? Male gametophyte.
Pollen grain.
Good. Excellent. What is the angiosperm female gametophyte? What is the name of the angiosperm female gametophyte?
Embryo sac.
Okay, very good. In the past, that's given us a little trouble. Sometimes when we've quizzed ourselves on that in the past, sometimes I think you've said that this was the megasporocyte or the megaspore, but hopefully now we've clarified that those come earlier in the process. Okay, very good. Now let's review what is involved. What is double fertilization? If there was an essay question on the test, what is double fertilization? What would you say?
When the two sperm of the generative cell fertilize with both the polar nuclei and the egg cell of the inside the embryo sac.
Good. If this was an essay question, you would probably want to mention what's the chromosome number of each sperm cell?
N.
And the egg cell?
N.
And what's the chromosome number of each polar nuclei?
N.
Very good. And then you would want to go on to say, what is the first product when the sperm cell... So what's the name when the sperm cell merges with the egg cell? What's the first product we get?
Zygote.
And what's its chromosome number?
2N.
What's the name of this process?
Fertilization.
Right. So those would be good things to make clear in an essay answer. In fact, it wouldn't hurt to even just write down a diagram just like I'm doing now as part of your answer. Okay, good. And what's the product of this process?
A triploid endosperm.
Excellent. What's the chromosome number for triploid?
3N.
Excellent. What's the function of the endosperm?
To store nutrients.
Excellent. For the growing embryo, what's the other structure in the seed that stores nutrients besides the endosperm?
The cotyledon.
That would be spelled cotyledon.
Oh, cotyledon.
Yeah. And what's another more name for cotyledons?
Scolium.
So this the scutellum is a type of cotyledon. That's right. The scutellum is a type of cotyledon and a certain type of monocot, I believe. What I was going though for is that a more common name for these are called the seed leaves.
Okay. Oh yes.
Or seed leaves. Okay. So what is a monocot?
Monocot is a type of plant with only one cotyledon.
In each seed. Good. And what's the other major type of plant that we've been studying?
Eudicot.
Yeah, eudicot. Good. And what's that?
It's a type of plant with two cotyledons in each seed.
Very good. Okay. Going back to the zygote, after the zygote has been formed, what's the very next thing that happens to it?
The zygote goes through one round of mitosis, and then it creates two cells, the terminal cell and the basal cell. The basal cell is the suspender, which kind of anchors the embryo, which the terminal cell becomes later on.
Very good. Now, what the term here is the basal cell. The basal cell becomes the suspensor. Suspensor. So an S, not a D. And then the terminal cell, as you were saying, is going to develop into the embryo. Okay. So let's continue with this process here. If you were trying to continue to describe the process, what would be some further steps for the embryo here?
The embryo becomes dormant in the seed, inside the mature plant.
Excellent. Very good. And then what would happen next?
The fruit or the seeds would be carried to like a different area or different location by either the animals that eat the fruit and then eliminate it, or by wind that may blow it to another direction.
Okay, good. So what's the part of the plant whose job it is to spread the seed, so to speak? Which part of the plant? I think you were already kind of mentioning this. I just want to be a little bit more specific about it. Which part of the plant is responsible for making sure the seed gets from one place to another? Maybe that's a weird question, but do you see what I'm saying?
No. Is it what? Wait.
So what I was going for is it's really the fruit's job.
Okay.
To make sure the seed gets from one place to another. For example, some fruit is tasty, so it attracts animals to eat it and then carry the seed somewhere else. Or some fruit has burrs in it, so it attaches to the fur of animals, and then they carry the fruit away, which carries the seed away. Or some fruit has little wings on it, so it gets carried away by the wind. So what's the job of the fruit? The job of the fruit is to get the seed from one place to another, one way or another. All right, so fruit. You kind of said that, but I just wanted to make that clearer. That's why. So it's easy to think that plants have fruit for our benefit because they grow fruit because we think that apples and oranges and grapes are tasty. But they're not doing it for our benefit. They're growing the fruit from an evolutionary standpoint to attract animals to carry the seeds from one place to another, or to make it easier for the wind to carry the seeds from one place to another. All right, so that's the job of the fruit. So the fruit is carried by wind or animal to a new location. Then what?
Well, once it's carried to a new location, when water, well, it'll still be on the ground, so it won't be growing until water actually flows into the seed.
Excellent.
For imbibition.
So that's an excellent point. For a while, the seed will probably remain dormant. And then what? Oh, so you already mentioned, then water
Imbibition
enters the seed. And that's good that you mentioned the name for that is imbibition. I don't know if you ever heard people talking about imbibing. Imbibing is a fancy word for drinking. So if people go to a party, they might say, oh, I imbibed a lot of alcohol there. So the seed here is imbibing water. Okay, then what? So this is the part that you were saying that your instructor specifically told you was likely to be on an essay question now on the next test. So let's make sure we have our essay answer ready for that. What would be the next step now?
After the water enters the seed, the hormone gibberellin is going to be given off by the endosperm. And then
which part of the seed releases the gibberellin?
The endosperm?
Actually, if you go back and review that slide, it's released by the embryo.
Oh, yeah. Okay.
That actually is maybe even, that could be more detail than your instructor would even require. It might be good enough just to say that the seed releases gibberellin. But if we're going to mention it, we should get it accurate. It's actually the embryo portion of the seed. Okay, but like I say, it might be sufficient just to say the gibberellin is released. Then what?
Once the gibberellin is released, the germination of the embryo will start.
Good.
And then it will begin to grow.
Good. So germination is initiated. Now let's be a little bit more specific. Remember, there was a specific slide with a series of steps that your instructor said was going to be tested directly. So what's the next direct step? What does the gibberellin directly cause to happen?
Gibberellin is going to cause the enzyme amylase to be released.
That's right. So now amylase is going to be released. Now, I think maybe we should be a little more specific. One thing, the actual name that's given on the slide for this is alpha amylase. So
Okay.
maybe that would be a better answer for the test. There's a couple different types of amylase. So we're actually going to be releasing alpha amylase. That's probably worth memorizing. It's in the slide. Okay, so the alpha amylase is released. It's released, I believe, by the outer covering of the endosperm. That might have been what you were thinking about when you mentioned endosperm in this part. So the amylase is released by the outer shell around the endosperm. Okay, what type of molecule is amylase? What type of molecule is that?
An enzyme?
That's right. What's the clue that would tell you it's an enzyme even if you'd never heard of it before?
The ACE
That's
at the
right.
end of a word.
Okay, so now what's the next step?
The enzyme is going to break down the molecules inside the endosperm, and that is going to create sugar. And then the sugar will enter the embryo, which will cause it for the roots to grow down and for the plant to start to sprout from the ground.
And for the shoot to sprout up. Excellent. Good. So the alpha amylase breaks down endosperm, forming sugar, which provides nutrients to embryo as roots and shoot begin growing. Okay, and I think that's as far as we'll take it right there. So this last part, so that's good. This last part, I think you said, was the part that was specifically likely to be tested. So this would be a good thing to just keep reviewing going up to the test. If you know something's going to be on the test, there's no reason not to have a well-prepared answer for that question. Okay, very well. Very good. So I think that's some excellent progress on that process. Again, one thing we didn't talk about yesterday that's probably worth having in your answer is that this is specifically called alpha amylase. Okay, good. One more thing that comes up a lot. What is the fate of the ovule?
That will become the seed.
That's right. And what's the fate of the ovary?
To become the fruit.
What's the purpose of fruit?
To disperse seeds to different locations.
Right. To disperse seeds by wind or by animals. Very good. By the way, you know, can you visualize like a pea, a green pea? So the fruit there is the what's this called? Is this called the pod? This would be the fruit. And then what do you call these inside? These are the seeds. These are the actual peas. So yeah, I guess this whole thing is called the pod. And I suppose each individual thing would be called a pea. So this would be an example then. So the pod here is the fruit.
And the pea is the seeds, okay.
One important thing to realize here is notice that biologists use the word fruit to mean something different than ordinary people. I think most ordinary people would not think of a pea pod as a fruit. When people think of fruit, they think of moist fruits like bananas and pineapples and apples and raspberries. But you should remember that to a biologist, there's also dry fruits. So this green pea pod would be considered a dry fruit. But in biology, it's still considered a fruit because its purpose is to be eaten by an animal so that they can carry the peas, the seeds, to a new location. Okay, very good. Okay, so here was a problem from your last quiz. How would you answer question 8 here?
During the process of germination, well, the hormone, during the process of gibberellin, basically the plant will start to grow. It's what causes the plant to grow. When water enters the seed during imbibition, the seed will release the hormone gibberellin, which causes the plant to go through germination, which allows the plant to grow into a mature plant. Hello?
Yeah, I'm here. Are you done?
Yes.
Okay, I think you probably wouldn't get full credit for that. I think you left out some of the steps we were just talking about. So you said that gibberellin was released, but I think they want more steps. So what would be the further steps?
Once gibberellin is released, the enzyme alpha amylase is released from the outer layer of the seed, and when that happens, the alpha amylase is going to break down the molecules in the endosperm to create sugar, and then that sugar will enter the embryo, which will cause it to grow.
Okay, that's a good answer. So this was a problem from the last quiz. So let's look at how we did the first time here. Germination is the process of adding the hormone gibberellic acid to the plant. So actually, so the name we've been using is gibberellin, but I think I said something incorrect last time. So the gibberellin is a type of acid, although gibberellic acid is only one of the gibberellins. It'd probably be better to say gibberellin here, but now I understand why they say GA, because gibberellic acid is a type of gibberellin.
Mm-hmm.
In any case, it says it's going to remove the plant growth from dormancy. I think it might be better to say the seed growth. And it causes fast, rapid growth. Now notice that in this problem we only got half credit the first time. So notice that they really are looking for those. Basically what happened here is you kind of stopped too soon in the answer. So that's why I wanted to review this. Notice they specifically said what they were looking for was the alpha amylase and then the sugars. So it's important to go through the whole process here. Also, they didn't mention it, but I think on the next exam it would probably be better to also mention the imbibition. That was something that got left out of here as well. So a couple of things that should be added to get the full credit are the imbibition and then leading to the alpha amylase and the sugar. Also, they didn't mark this as wrong, but the next grader might mark this as wrong. Take a look at what we wrote here. An example of this is when grapes are treated with gibberellic acid, and the ones treated with gibberellic acid grow more quickly. Can you see why I would say that's not a good example for this particular question? It's true that gibberellins do cause fruit growth. It's true that gibberellins do cause fruit growth, but why is that really not appropriate to include in this answer?
Because it's not part of, it's not necessary, and he just wants to know what happens during germination, not what they found using the hormone.
Good. Now, is fruit growth a part of germination?
Seed growth and stem growth are.
So is fruit growth a part of it?
No.
No. So that's my main criticism of that. So the next grader might take off credit for that. It's true that gibberellins do cause fruit growth, but that's a totally separate process from germination. Remember we saw up here, first the fruit is carried by the wind to a new location, and only later, after dormancy, are we actually going to start germination. Germination is initiated later. So those are two separate processes. Okay, so anyway, it looks like that was a question. So basically, it looks like this question was already on quiz 2, and it looks like the instructor basically warned you they're going to ask basically the same question on the next exam. So you're prepared for that. Remember, so the question might be worded as describe the process of germination, or it might be worded as describe the process by which nutrients are mobilized for the growing embryo. If they ask you how nutrients are mobilized, that would basically be the same question.
Mm-hmm.
Good.
Oh, yeah.
Okay, well that's a good recollection of the things that we were talking about last time. So it's clear that you were carefully reviewing that, so that's good. I think that's excellent progress, and it's good you're reviewing that previous material. These videos are offered on a pay-what-you-like basis. You can pay for the use of the videos at my website. There's a link to my website in the annotation. The address is www.freelance-teacher.com/videos.htm. Or you can just use the link in the annotation. Thank you.